and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Welcome, everyone, to our Pharmaceutical Quality Symposium 2023, Quality, Supply Chain, and Advanced Manufacturing. Good day, good afternoon, or good night, depending on where you may be in the globe. It is extremely exciting to see so many of you online with us here today, and I actually recognize some names from previous attendees and previous conferences. We certainly appreciate your commitment in attending this event and meeting any scheduling challenges that you may have had. We invite you to chat amongst yourselves in the chat pod when it is available during the breaks. And from this morning's conversations, I see that we already have significant representation, global representation, and that, of course, is a total encouragement to see such lively participation. As many of you may know, my name is Brenda Stoddard, and I'm with the Small Business and Industry Assistance Program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. It is my extreme and great pleasure, along with my colleagues, to be with you here today for what is our third biannual Pharmaceutical Quality Symposium. We started in 2019. It is a definite privilege to be able to collaborate once more with our Office of Pharmaceutical Quality to bring you this two-day event, which is saturated with insight on topics related to pharmaceutical quality regulation, supply chains, and advanced manufacturing technologies. We entreat you to make the most of this experience, attend all the sessions that interest you, learn everything that you can, submit your questions during the available Q&A sessions, and network in the chat pod. In addition to the Adobe Connect platform, we are offering live YouTube streaming, so please go ahead and invite your colleagues. However, please note that only those attendees who register and attend via Adobe Connect and who log into Room 1 will be able to submit questions to our speakers. And now, as we set up for this poll, which I try to do each conference, many of you know, yes, I can see the whole screen. Some of you have been with us for quite some time, even going back as far as 2010 when the program really started. But I dare say that many of you are also first-time attendees, and a special welcome to you as we work towards expanding our audience base and increasing our portfolio of SBIA events. And we depend on you attendees to spread the word by mouth as well of our offerings. So to break the ice and get us thinking, we have the following question, and many of you have already, oh my, nobody's chosen E. Many of you have already checked the box. So the question is, how many SBIA conferences have you attended? And your choices are A, this is my first one, B, 2 to 5, C, 6 to 10, D, more than 10, and E, most since 2010. All right, let me give it a few more seconds here because I think we've already stabilized our votes. All righty then, so 36% indicated that this is their first event. Welcome, a very hearty welcome to you, and I hope you will be with us throughout your career. And 1%, I'm glad I waited. 1% indicated that they attended almost all since we started in 2010. So thank you very much for your loyalty. Thank you guys for participating. Okay, as of this morning, we have over 4,100 registrants representing 114 countries. Now, over 51% of these registrants have self-identified as small businesses, and of those, over 21% report having 1 to 9 employees. As you can imagine, these stats continue to be extremely reassuring to us here at CEDAR and SBIA, in that they confirm that we are indeed reaching the small business sector of our audience, and they also validate our mission and the very program itself. What you see here is a snapshot of our geomap for today's event. And this map is always quite exciting to me personally, also to the planners and the operating divisions here at FDA, for it provides a very powerful visual as to the impact and extent of our outreach. And I usually have fun with it, and you can do the same. You can actually click on the image and it will take you to your browser where you can zoom in and have a closer look at our global participation. So try it, zoom in on your geographic area and see if anyone else is attending from your city. This here, of course, is a slide that provides the QR codes for social media avenues, and you can scan them with your phones and be taken to the appropriate platform. They will also be advertised across the screen banner during the breaks if you miss it now. So let's turn our attention to some logistics. Again, as you know, this event is 100% virtual, and over the course of these 2 days, we will have a total of 26 speakers and panelists, not to mention our very own organizers and technical staff, all working from our own locations. And as such, there may be some internet issues, but please bear with us. And if in fact we encounter those, we will get them resolved as quickly as we can. And speaking of tech issues, here is a link for a one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. It's the same page that you used this morning for your online access, so please bookmark that page as you will probably need it over the next 2 days. And if you have any technical questions, please check there first, as you will most likely find a solution there. We invite you to submit your questions to us so that we can have a vibrant and productive Q&A segment, which is so crucial to these events. And I can't emphasize enough that this is a learning opportunity. That's the whole mission of SBIA, to provide learning resources and education. So I encourage you to take full advantage of the expertise we offer here. We will entertain as many questions as time allows. And when you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name, as shown here. Also, during each presentation, there is a link on the right side of the pod where you will see the speaker's headshot, and there's a link there that says download the slides. You can definitely do that. But note that this link is only active during the real-time presentation. In the same area, there will also be a link to a speaker evaluation, and we encourage you to take the time to complete that evaluation, as your feedback is very much appreciated by the individual speakers. In addition to the speaker evaluation, we have our final event evaluation or survey. We use the terms interchangeably. Upon completion of this survey, an attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. The trick is that the certificate must be downloaded immediately upon completing the survey, otherwise that opportunity is lost to you. Now, even if you do not want or need a certificate, please take a few minutes to complete the conference evaluation, as your feedback will help guide the design of our future events. Note that both the evaluation and certificate are only available for 2 weeks, which means that the deadline date is November the 15th. The survey link will be posted on our one-stop shop page this afternoon. And as a reminder, all the sessions are being recorded. It will take us a week or so a week or two, to process the recordings, but you should soon be able to access them from our SBIA's homepage. And I hope that you have already bookmarked that page, which is fda.gov/CDERSBIA. In the interim, the YouTube link for both days will be posted on the One Stop Shop webpage at the end of the day. To simplify things, SBIA will send our thank you email to all the Adobe attendees tomorrow post-event, and that email will also have the survey link should you miss it over the next 2 days. Now let's take a look at our upcoming events. November 16th, we will host our webinar, Common Issues with SEND Data Submitted for Safety Pharmacology Studies. From the information provided in this webinar, industry will be able to apply this knowledge to update their systems to avoid future errors and so increase usability of the SEND data sets for regulatory reviewers. Then on November 28th, we will have a webinar toward global IDMP implementation, a focus on global use cases. Here we will discuss updated data quality information, focusing on submitted study data conformant to the Standard Exchange for Non-Clinical Data Implementation Guide version 3.1. December 6th through 7th, we will host our FDA Clinical Investigator Training Course 2023. And this course is specifically designed to promote professionalism in the clinical trial industry for individuals involved with FDA application submissions, and also to familiarize stakeholders with the regulatory and science issues involved in the development and approval of medical drugs and biologic products. And then let's move on to 2024. This is a personal invitation to join us for this hybrid meetings as we resume our in-person component. On February 13th through 15th, we'll be hosting a joint US FDA, MHRA UK, Health Canada Good Clinical Practice and Pharmacovigilance Compliance Workshop. It would be so wonderful to have that energy of having a room filled with attendees once more. Please, if you can make it, try. It's going to be fantastic. Just the in-person dynamic. This 3-day workshop will focus on global clinical trials and good clinical practice, bioequivalence, and pharmacovigilance in the post-pandemic world. Check our homepage, fda.gov/CEDAR-SBIA, seen here for upcoming events. And we also invite you to visit our CEDAR Learn webpage, where you can link to SBIA tutorials, along with other educational resources, many of which offer healthcare professional CEs. And that website is fda.gov/CEDAR-Learn. This slide here calls attention to a short video which will give you an idea of our SBIA resources. I will not play it now in the interest of time, but it will be available to you to play during the lunch breaks. Now, at this point, I would love to acknowledge those individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit as well. A special thanks to Kimberly Monson and Erin Wood, who are responsible for designing the agenda, recruiting the speakers, and putting together all the symposium contents. So thank you so much, Kimberly and Erin. But most of all, our heartfelt gratitude to you who took time out to attend this event, regardless of the inconvenience of the various time zones. And we do not take this commitment lightly, for we would not be here if it were not for you. A very special recognition to my colleague, Captain Ray Ford, who will be your host during the next 2 days. And now I am pretty sure you are ready for us to launch fully into the day's agenda. As a reminder, our speaker bios are all available in their totality on the One Stop Shop website. But for now, I am honored to introduce our 2 keynote speakers. Delivering our FDA keynote is none other than Dr. Robert M. Califf, who is the Commissioner of Food and Drugs. Previously, Dr. Califf served as Commissioner of Food and Drugs from February 2016 to January 2017. As the top official of the FDA, Dr. Califf is committed to strengthening programs and policies that enable the agency to carry out its mission to protect and promote public health. Dr. Califf served as the FDA's Deputy Commissioner for Medical Products and Tobacco from February 2015 until his first appointment as Commissioner in February 2016. Following Dr. Califf, we have our very own OPQ, Dr. Michael Kopcher, who will give the Office of Pharmaceutical Quality's keynote. Dr. Kopcher is the director of the FDA's Office of Pharmaceutical Quality, and this office has over 1,300 staff responsible for assuring the availability of quality medicines for the American public through assessment, inspection, surveillance, research, and policy. OPQ contributes to the assessment of nearly every type of human drug marketing application, including new drug applications, abbreviated new drug applications, and biologics license applications, including 351(k) applications, aka biosimilars. OPQ also performs the quality assessment of investigational new drug applications and establishes quality standards for over-the-counter drug products and facilities. Please, let us welcome our keynote speakers.
The FDA's mission to protect and promote public health is wide-ranging, covering an enormous array of areas of oversight and responsibilities. One of the most critical of these responsibilities, and the focus of today's symposium, is ensuring that drug products marketed in the U.S. are safe, effective, and of high quality. American patients and consumers deserve to be confident in the safety, efficacy, and quality of the drug products they take. And the FDA works diligently to ensure this reliability and trust in our processes. Each of these areas of focus comes with its own challenges, both in terms of defining standards and meeting them. The concepts of safety and efficacy are complicated and require extrapolation from the clinical trials that comprise the body of evidence required for approval for marketing. However, the subtlety and multifaceted nature of product quality is often underappreciated by the public and definitely underappreciated by many academics who focus only on the clinical data. It's quality that ensures a patient can trust their next dose as much as their last. Today's symposium will focus on this aspect of the FDA's work and shed additional light on the tools and methods we employ to make this assurance possible. These tools include the quality assessment, developing and producing policy to guide industry, supply chain surveillance, and staying at the forefront of emerging and advanced technologies. You'll be hearing about each of these parts of the process in great detail during this meeting, so I'll just briefly discuss these pieces and how they fit together. The core of the quality equation involves the assessment we make in the initial application. It's a pivotal part of our rigorous approval mechanism. When we conduct a quality assessment, it means we're going to take a deep dive into reviewing the manufacturing processes, microbiological issues related to product quality and manufacturing, as well as facilities issues. This part of the process also includes thorough evaluations of the drug substance, the drug product, and biopharmaceutics portions of the product application we receive. The goal of this oversight is simple and straightforward, ensuring the consistent availability of safe and effective medications. By identifying and communicating potential quality issues early on, we empower the industry to correct these issues, thus reducing the risk of recalls or shortages, and improving the chances that we will avoid shortages due to later quality problems as we work to ensure that we will have a supply of quality drugs for the patients who need these drugs. It's a win-win for all involved. We strive to make the FDA the global benchmark for the regulation of pharmaceutical quality. This not only means ensuring that the products themselves are safe, effective, and of good quality. It also means that this standard is built into the production and development process. In other words, it means that it is part of our efforts to foster an entire culture of innovation that includes a focus on quality. To this end, the FDA has pioneered integrated quality assessments and more recently refined our aligned teams approach, in which a team of experts evaluates the respective parts of the application in a collaborative environment. As we continue to evolve, we're developing new and even more effective approaches to achieving these ends. For instance, we have spearheaded the Knowledge-Aided Assessment and Structured Application, or so-called CASA initiative, which moves from a narrative format to a structured, data-driven risk assessment method. Beyond the assessments themselves, we also play a pivotal role in shaping industry policies, providing beacons to help guide the pharmaceutical industry in their work to guide towards ensuring safety, efficacy, and quality. Policy initiatives include regulations, of course. We're a regulatory agency, and regulatory science is a big part of our overall plan. Knowledge in pharmaceutical quality is not static. But these efforts extend beyond simply regulations. They can serve as effective communications with and guideposts for the industry and to support the production of drugs that consistently meet our rigorous criteria. These tools can include guidance documents, international collaboration, compliance programs, and dynamic regulations. The goal is to use this assortment of communications so that we can ensure everyone has the opportunity to understand our expectations so that no one has to play catch-up. Setting thoughtful policy is critical because this is one of the best ways to communicate our expectations prior to the submission of a drug product application to the FDA. It also ensures that applicants are on a level playing field and have access to the same information wherever they are in the drug development cycle. A testament to the efficiency and efficacy of our policy was our rapid response during the COVID-19 crisis, providing timely guidance on vital topics, ensuring drug availability and quality, even during unprecedented challenges. I was not at the FDA as the pandemic crisis unfolded, but since rejoining, I remain in awe of what was accomplished. Guaranteeing drug quality doesn't end at approvals. We actively monitor supply chain integrity, and we are taking steps to do all that we can to keep every drug consistently available and compliant with current good manufacturing practice. Of course, the quality of a drug that's manufactured is of little value to a patient who cannot access the product because of shortage. We've been troubled by shortages going back over a decade, but the pandemic was both an eye-opener and a catalyst for public and political awareness of the issues we face relating to shortages and other supply chain vulnerabilities. We're committed to fostering an environment conducive to strengthening these supply chains, all the while ensuring drug quality. We're working on many different fronts to address these challenges, and we'll need collaboration across government and with the private sector to reach a point that shortage becomes a rare thing. This is especially important as more and more life-saving drugs reach generic status. A major gift to the public health for all 340 million Americans, but also for the other 7.6 billion people in this world. In addition to our technical and quality work, we're hopeful that the economics of generic drugs will undergo reform to optimize investment in quality systems. One way the FDA is working to shore up supply chains and provide a basis for needed investment is through developing a quality management maturity program, which will incentivize drug manufacturers to voluntarily invest in quality culture. What quality management maturity really means is a state in which drug manufacturers have successfully integrated their business and manufacturing operations with quality practices and technological advancements to optimize product quality, enhance supply chain resiliency, and drive continual improvement. In other words, manufacturers have developed a culture of quality within their own organization. The FDA has already completed 2 pilot programs on quality management maturity from 2021 to 2022. And currently, we're using the lessons learned to help develop a program that will incentivize industry to voluntarily work towards fostering a strong quality culture mindset. We also continue to evolve as the industry does. Anticipating and adapting to technological advancements is crucial to sustaining and promoting a culture of quality. This means understanding tomorrow's innovations today, particularly those involving advanced pharmaceutical manufacturing technologies. These are technologies that will provide more flexibility and agility to the manufacturing process and enable more timely responses to urgent health issues than traditional manufacturing technologies and facilities. We need to understand these technologies and how to regulate them. Our emerging technology program encourages early industry collaboration with the FDA, facilitating transparent dialogues for those employing pioneering manufacturing and testing methods. We also have established the framework for regulatory advanced manufacturing evaluation, also called FRAME, which works to provide clarity and reduce uncertainties for products manufactured with advanced technologies. The goal is to develop a framework of robust regulations to ensure that industry has clear guidance on how to apply advanced manufacturing to their products, such as applying artificial intelligence and continuous manufacturing to the pharmaceutical industry. FRAME will ensure that as the industry progresses, we're right alongside, ready to support and guide. Over the next 2 days, you'll have the opportunity to collaborate on an exploration of the true meaning of pharmaceutical quality. Together, you'll consider and envision a future where the highest drug quality standards are met with minimal regulatory burden. Your partnership in this effort is invaluable, particularly at a time when we face challenges posed by those spreading misinformation and seeking to undermine confidence in the science on which we reach our decisions and the quality of the industry as a whole. I'm confident that working together, we can enhance trust in science, the industry, and the FDA, even as we strengthen and reaffirm our commitment to ensuring the American people have access to the safest, most effective, and highest quality medications. Thank you.
Thank you, Dr. Califf, and thank you all for coming today to discuss some of the important aspects of pharmaceutical quality. I am Mike Kopcha, and I'm the Director of CDER's Office of Pharmaceutical Quality, or OPQ. As Dr. Califf has underscored, American patients deserve unwavering confidence in their drug products. The mission of OPQ is to assure that quality medicines are available to the American public. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality, then, is what assures the availability, safety, and efficacy of every dose. Over the next 2 days, here at the Pharmaceutical Quality Symposium, you'll be hearing about the essential pillars for pharmaceutical quality, which include quality assessment, quality policy, supply chain, and advanced manufacturing. These pillars are the symposium's session topics, and our presenters will describe our ongoing efforts to assure pharmaceutical quality for the American public. We will kick off today's first session with quality assessment, an essential part in the work that we conduct in OPQ. OPQ's quality assessment approach integrates multidisciplinary insights by utilizing science-driven, risk-based standards to assess regulatory submissions. You'll hear a very comprehensive description of how OPQ is working to advance the quality assessment process with new technology. In addition, we will also touch on patient-focused specifications that incorporates the patient's voice in drug development and evaluation. This is the primary goal of patient-focused drug development. By considering multiple factors, including patient-focused specifications, we are reshaping and reforming the drug evaluation paradigm. Our innovative strategies, as well as the collaboration between regulators and industry, facilitated the approval of many generic drugs, maintaining an impressive on-time action rate during challenging times. As regulators, there were many lessons learned that have increased our agility and response to an evolving world, and one area of focus is applying those lessons to reshape the future of inspections. You will hear some common deficiencies observed in pre-license inspections, or PLIs, for CETA-regulated products to highlight critical areas to consider for improving quality operations at facilities. Today's afternoon session will cover quality policy. Beyond assessment, policy is a potent tool in our toolbox. Through guidance and other policy documents, we convey the FDA's perspective and thinking on pharmaceutical quality topics. In a complex global landscape, we engage internationally via the International Council on Harmonization, or ICH, issuing guidelines and guidances such as the transformative ICH Q12. You will hear about FDA's recently released guidance to address nitrosamine drug substance-related impurities, or NDSRIs. Domestically, collaborations like the one between FDA and the United States Pharmacopoeia play a pivotal role in developing and upholding quality standards. Product-specific guidances, or PSGs, from the Office of Pharmaceutical Quality and the Office of Generic Drugs offer valuable insights for drug manufacturers and exemplify our cooperative approach to policy. Tomorrow we'll start the day spotlighting Project Orbis, which was initiated by FDA's Oncology Center of Excellence. Project Orbis provides a collaborative framework for concurrent submission and review of oncology product applications with other international regulatory agencies. Then we will deep dive into supply chain quality, which concerns many Americans. Supply chain quality issues are one of the root causes of drug substances. In a recent analysis at FDA, what was shown that 46% of drugs newly in shortage were due to quality issues. I've said before that I wish my job wasn't necessary and that all manufacturers could simply be trusted to make quality products. Unfortunately, we must remain vigilant globally, which you saw in our swift response to the concerns raised by the World Health Organization, or WHO, over pediatric medicines contaminated with diethylene glycol and ethylene glycol, which killed children in the Gambia. Thankfully, U.S. products were not impacted. But what could have happened if we were not vigilant? We remain vigilant against contamination of all kinds, including from microbial sources. In fact, we have a guidance available to assist manufacturers of active ingredients and finished dosage forms with establishing and meeting microbial quality standards for non-sterile drug products. Our Quality Management Maturity, or QMM program, encourages drug manufacturers to implement quality management practices that go beyond current good manufacturing practice requirements. In addition to QMM, CEDAR's drug quality sampling and testing program and site selection model help us maintain surveillance of drug quality. Remember, pharmaceutical manufacturers, regardless of their location, have a duty to ensure that the products reaching U.S. patients are of high quality. The final session of the symposium discusses the potential benefits of advanced manufacturing in the pharmaceutical industry. Advanced manufacturing is, in part, using novel technologies to improve the pharmaceutical manufacturing process and better assure quality medicines are available to patients and consumers. FDA's Emerging Technology Program, or ETP, reduces hurdles for potential applicants by providing a forum for stakeholders to engage in early dialogue about advanced manufacturing technologies with regulators. In concert, CEDAR's FRAME initiative aims to provide clarity and reduces uncertainty for developers of products manufactured with advanced technologies. An example of delivering clarity for stakeholders can be seen with the implementation of the ICH Q13 guidance on continuous manufacturing of drug substances and drug products as a final FDA guidance. CEDAR's intramural as well as extramural research program has several projects focused on continuous manufacturing, some which include applications of artificial intelligence or AI. You will hear more about AI's role in drug manufacturing as we seek to leverage the advantages of AI for patients and consumers while proactively protecting them from any associated risks. Remember, we work on behalf of U.S. patients and consumers. In closing, maintaining the integrity of pharmaceutical quality requires an all-hands-on-deck approach. It's imperative that FDA, along with our stakeholders, use available resources to assure patients have access to quality medicines. And that brings me to my last remark. Regulators or industry cannot assure quality by working separately. Pharmaceutical quality requires our data-driven, science-based collaboration. Thank you for the privilege of your time, and please enjoy the next 2 days of the symposium. Your next speaker is Dr. Neil Steiber, who is the Associate Director for Science and Communication in the Office of Quality Surveillance, who will discuss the state of pharmaceutical quality.
Good morning. My name is Neil Steiber, and I work in OPQ's Office of Quality Surveillance. It is my pleasure to discuss the state of pharmaceutical quality with you today. We will seek to better understand pharmaceutical quality by looking at OPQ's annual report on the state of pharmaceutical quality. In this talk, I'll share with you the background for the report on the state of pharmaceutical quality. We will quickly survey some highlights from each of the past 5 reports. Then, in a bit more detail, we will discuss the most recent report on the state of pharmaceutical quality, the fiscal year 2022 report. Lastly, we'll look forward to the report on the state of pharmaceutical quality for fiscal year 2023. To advance OPQ's vision and the themes of today's symposium, I would like to emphasize that reliable supply chains provide quality drugs when and where patients need them. This concept is at the core of the report on the state of pharmaceutical quality. Let's start by talking a little bit about the background for the report on the state of pharmaceutical quality. By providing the public with information on the quality of drug manufacturing sites and their products, the report on the state of pharmaceutical quality supports OPQ's mission and the mission of my sub-office, the Office of Quality Surveillance. The report on the state of pharmaceutical quality has been published annually since fiscal year 2018. All of these reports are available on the website, and you can find this website and all of those reports simply by searching for the report on the state of pharmaceutical quality. Each report on the state of pharmaceutical quality looks at indicators of quality. These indicators include site inspections, regulatory actions, post-market quality defect reporting, sampling and testing results, recalls, import alerts, and more. The report on the state of pharmaceutical quality analyzes these data to identify trends and insights that provide lessons for FDA and for you, the external stakeholders. The report on the state of pharmaceutical quality also includes a section on commitment to quality that describes programs and tools that will improve quality surveillance in the future. The first report on the state of pharmaceutical quality was issued for fiscal year 2018. It set the foundation for the types of analyses that would become the report on the state of pharmaceutical quality. One emphasis has been demographics. As shown by the pie charts on the right, these data help to answer the questions: where are manufacturers located? What types of manufacturing, whether generic, brand, or non-application products, are they engaged in? Another emphasis of the reports on the state of pharmaceutical quality has been the analysis of product quality defects, as seen by the bar graphs in the middle of this slide. For the fiscal year 2019 report on the state of pharmaceutical quality, we built on those same topics and sought to more deeply understand how inspection outcomes vary by geographic regions, application types, and manufacturing sectors. That is shown by the figure on this page. We also provided results of drug product sampling and testing. And in addition, this report for fiscal year 2019 shared a deeper dive into the citations from CFR 211 that are referenced in FDA 483 forms that describe violations that have been observed during inspections. For the fiscal year 2020 RSPQ, we analyzed the causes for some of the larger recalls over the prior few years. We sought to answer what drove these recalls. One major cause of recalls, which can be seen by the black and gray lines in this chart, was nitrosamine contamination. The presence of this carcinogen led to the recall of ARBs that are used to treat high blood pressure. Nitrosamine contamination also led to the recall of ranitidine that is used to treat and prevent stomach ulcers. This graph also shows that in 2020, with the beginning of the COVID-19 public health emergency, we started to see the recall of alcohol-based hand sanitizers due to methanol contamination. Challenges with alcohol-based hand sanitizers continued into fiscal year 2021. The report on the state of pharmaceutical quality for fiscal year 2021 showed the rate of non-compliant products from FDA's sampling and testing. During fiscal years 2019 and 2020, there was a large increase in non-compliant results. These were for impurity testing and were due to products containing nitrosamines. Then, during fiscal year 2021, there were many non-compliant results for both assay and impurity testing that were due to alcohol-based hand sanitizers. It is important to note that many of the non-compliant hand sanitizer products were from products that were manufactured at newly registered facilities after FDA issued temporary guidance during the COVID-19 public health emergency. Now let's move to the fiscal year 2022 report on the state of pharmaceutical quality. This table shows the location of drug manufacturing sites for countries with at least 50 sites. The CEDAR site catalog includes all manufacturers of application and non-application products. However, it's important to note that the CEDAR site catalog does exclude several types of sites which are managed separately. For example, it excludes medical gas manufacturers. It also excludes registered outsourcing sites under 503B of the Federal Food, Drug, and Cosmetic Act. And lastly, it excludes manufacturers that only produce alcohol-based hand sanitizers and who registered during the COVID-19 public health emergency. These types of sites are managed separately and are not included in the CEDAR site catalog. As you can see from this chart, in fiscal year 2022, there were 4,814 drug manufacturing sites in the CEDAR site catalog. That is an increase of 12% from fiscal year 2018. Of interest, 40% of these sites are in the no application sector. That indicates that all products manufactured at those sites can be legally marketed in the U.S. without an approved FDA application. Continuing with the fiscal year 2022 report on the state of pharmaceutical quality, let's talk a bit about what we call essential medicines. First, let's define essential medicines. Pursuant to Executive Order 13944, which was issued in August 2020, FDA established, a few months later, a list of 227 drug and biological products, which are known as the List of Essential Medicines, Medical Countermeasures, and Critical Inputs, or in short, it's called Essential Medicines. Included in this list of essential medicines are 168 CETA-regulated drugs. This chart shows that there is a heavy reliance on foreign manufacturing for FDF, and especially for API. In addition, the report on the state of pharmaceutical quality shares that 48% of essential medicine products have at least one domestic API manufacturer, and nearly 90% of essential medicine products have at least one domestic FDF manufacturer. It is important to note that this analysis is based on counts of manufacturing sites because manufacturing volumes remain uncertain. Now let's discuss import alerts and continue on the fiscal year 2022 report on the state of pharmaceutical quality. This chart shows the 28 import alerts that FDA issued during fiscal year 2022 for reasons related to drug quality. Based on the report on the state of pharmaceutical quality, I would like to share a few observations about these import alerts. First, 89% of these sites that were placed on import alert produced non-application, non-sterile finished drug products. Secondly, most of those import alerts were related to drug quality for sites that manufacture hand sanitizer. It is important to note the role that FDA's authority for record requests under the Food, Drug, and Cosmetic Act, Section 704A4, has on import alerts. Many of these import alerts resulted from those record requests. In particular, the column on the right shows that 10 of these import alerts were for sites that refused to respond to Food, Drug, and Cosmetic Act 704A4 record requests. Also, the second column from the left shows that 4 import alerts were due to current good manufacturing practice, CGMP deficiencies, that were identified through these record reviews. This demonstrates the ability of record requests and their review to identify sites with deficiencies in CGMP compliance and those sites that are unwilling to provide evidence of CGMP compliance. Another key observation from this chart is that the number of import alerts for sites in China and South Korea are disproportionately high. China and South Korea had 43% and 36% respectively of these import alerts, yet they represent only 15% and 4% respectively of foreign sites in the catalog. The vast majority of the import alerts related to drug quality for China and South Korea, 86%, were for hand sanitizer manufacturing sites. Continuing our discussion about the fiscal year 2022 report on the state of pharmaceutical quality, let's look at recalls. This chart shows recalls over the past 5 years for specific quality-related defect groups. In fiscal year 2022, 166 sites generated 912 recalls. This was the highest number of recalls in 5 years. As in prior years, the largest defect group for recalls continues to be CGMP deviations, and this is shown on the chart by the light blue bars. Of interest, during fiscal year 2022, there were 4 specific events that were associated with a total of 37% of all recalls. These 4 events included temperature abuse for products in storage, a contaminated excipient, and other CGMP deviations. Additional specific information about those recalls are available in the fiscal year 2022 report on the state of pharmaceutical quality. Now I would like to share some insights about warning letters from the fiscal year 2022 report on the state of pharmaceutical quality. This chart shows warning letters that were issued over the past 5 years by region or country. In this chart, the U.S. is in black at the top of the bars. In recent years, the share of warning letters issued to U.S. sites has varied. For fiscal year 2022, there are a few key observations. First of all, 68% of these sites for fiscal year 2022 were FDA manufacturers of non-sterile, non-application products, even though these sites represent only 30% of all fiscal year 2022 inspections. The second observation is that 31 warning letters were issued to hand sanitizer manufacturers. 14 of those were issued solely based on methanol impurities that were detected during FDA's product testing. For fiscal year 2022, this chart shows an increased proportion of warning letters issued to domestic manufacturers as compared to the prior 2 years. This was due to the ongoing COVID-19 public health emergency and related travel restrictions. During fiscal year 2022, a high percentage of inspections were domestic, and that corresponded with a higher percentage of warning letters being domestic. Also of interest, going back to fiscal year 2021, we see that there were a large number of hand sanitizer-related warning letters issued to sites in Latin America, that is shown in yellow. These sites were primarily in Mexico. Details about FDA's warning letters are available through a searchable database of warning letters, and information on that can be found via the report on the state of pharmaceutical quality. The report on the state of pharmaceutical quality also provides updates on programs that FDA has developed and is developing that demonstrate our commitment to quality. These programs will improve our ability to provide quality insight of site and product quality. In particular, the NIP is improving inspections by developing IT systems to collect and manage inspection data. Quality management maturity is seeking to strengthen quality management practices and promote supply chain reliability. And the Coronavirus Aid, Relief, and Economic Security Act of 2020, also known as CARES, requires reporting of amounts manufactured. These data will strengthen drug quality surveillance. With these advances altogether, FDA will be able to improve inspections and quality assessments to enhance supply chain reliability. It will also help to reduce drug shortages and bolster emergency response. These enhanced risk management tools will enable FDA to better prioritize quality surveillance resources and thereby protect the public from non-compliant products. Overall, we believe that this will increase confidence in the supply and quality of marketed drugs. Lastly, from the fiscal year 2022 report on the state of pharmaceutical quality, let's discuss amount reporting under the Coronavirus Aid, Relief, and Economic Security Act of 2020, also known as CARES. This chart shows the percentage of products for which amounts have not been reported under CARES. The message from this chart is clear. The amount manufactured has not been reported for the vast majority of products across product types. FDA hopes the reporting rates will increase and intends to use CARE's drug amount data to support drug quality surveillance in a few important ways. First, CARE's amount data will enable us to better approximate market share. Secondly, with CARE's amount data, we can gain insights on vulnerabilities and dependencies in the drug supply chain. Third, these data will support responses to natural disasters, supply chain interruptions, and potential shortages by identifying alternative manufacturing sites. And lastly, the CARES amount data will help to strengthen the site selection model and better prioritize surveillance inspections by providing an important proxy for exposure to drug products. And now, looking ahead, we are currently planning for the fiscal year 2023 report on the state of pharmaceutical quality. As in the reports that we have already discussed, this upcoming report will continue to assess quality-related data, including product recalls and product quality defect reports, to identify trends in site quality and product quality. We will continue to use cutting-edge and innovative analytics to analyze data on regulatory actions and product quality testing for insights that can be of value for us inside the agency and can be useful to you, our external stakeholders. We also plan to share progress about the development and implementation of programs that can better characterize pharmaceutical quality and help assure the availability of drugs. Thank you very much for your time and attention during this presentation. I hope that you've learned a lot about the report on the state of pharmaceutical quality. And if you're interested to learn more, please look up the reports, which are all available on the web. Thank you very much.
Thank you for that update, Dr. Steimer, on the state of pharmaceutical quality. This is Ray Ford serving as your host for the remainder of the day's events. Next, we'll discuss quality assessment updates by Dr. Larry Lee, who serves as the Deputy Director of Science in OPQ. Our final presentation in this session before the break is on patient-focused specifications by Dr. Susan Kirshner, the Division Director in the Office of Biotechnology Products and OPQ. For a complete biography on all presenters and panelists today and tomorrow, please consult the speaker's biography located in the Agenda and Speaker Bio section that you may have noticed during registration. Now, please join me to welcome Dr. Lee.
Good morning. I'm going to talk about our effort in advancing our quality assessment to meet the challenges of increasing volume and complexity of regulatory submissions. It is important to first talk about the importance of pharmaceutical quality. In general, a quality product means that it consistently meets the expectations of the user. Drugs are no different. To understand the importance of pharmaceutical quality, it is necessary to relate pharmaceutical quality to patients' perspective. Specifically, patients expect safe and effective medicines with every dose they take. Pharmaceutical quality is consistently meeting standards and ensure every dose is safe and effective, free of contamination and defects. It is what gives patients confidence in their next dose of medicine. The Office of Pharmaceutical Quality, OPQ, within CDER, oversees the quality of new drugs, generic drugs, biologics, biosimilars, and over-the-counter drugs. All of drugs applications have a quality, or the so-called chemistry, manufacturing, and controls CMC section. One of the core functions of OPQ is to assess this session. Specifically, our assessors evaluate how product is designed, how it is manufactured, and the manufacturing facilities in order to ensure a safe and effective drug is being delivered consistently to intended patient population. OPQ also assesses product and manufacturing changes after a drug is approved to ensure quality is maintained. Although there have been noticeable improvements in the drug development and manufacturing arena, regulators are still facing challenges in assessing quality information in drug applications. The number and the complexity of the applications have increased significantly in the past few years. And at the same time, we are tasked to evaluate these applications in a shorter time with same resources. Every year, at OPQ, we evaluate on average more than 3,000 INDs, more than 200 new drugs and biologics applications, more than 900 generic drug applications, and more than 10,000 post-approval changes. One problem is that we receive all these applications as unstructured information in PDF files. As a result, our assessments are freestyle, unstructured narratives, in which a significant portion of the documents are summarization of information or copy and paste data from the submissions. Such a system poses problems because the risk assessment and evaluation of the applicant's mitigation approaches get dispersed in lengthy narratives. Oftentimes, there is inconsistency and ineffectiveness, as well as difficulty to share knowledge and manage FDA's information on approved drug products and facilities. Our decision-making capacity may not be optimized because assessors evaluate each application in relative isolation without fully assessing the wealth of information at FDA disposal. We soon realized that good knowledge management is essential. In the context of technology advancements, we cannot continue to assess quality through our traditional narrative-based evaluations, using unstructured tests, summarization of application information, and copy and paste data tables. I would like to point out that these practices do not allow for easy knowledge sharing, management of quality across product life cycle, and overall modernization of assessment. Instead, in order to be most efficient, we want to take advantage of modern IT tools and platforms and emphasize structured data and the ability to capture critical information. This will move to highly specific, stored in a predefined format structured data, which will enable us to achieve a systematic approach to risk assessment, resulting in a more consistent, high-quality evaluation and decision-making. And the idea here is based on efficient information exchange, knowledge management, and data analytics. At OPQ, we recognize the need to modernize our quality regulatory assessment, and we are currently taking steps to transform our evaluations from narrative information to structured data and systematic approach for risk assessment. powered by IT, so we can best capture and manage knowledge. This concept was envisioned in 2016 and discussed at the Pharmaceutical Science and Clinical Pharmacology Advisory Committee meeting on September 20th, 2018, as CASA, an IT system that modernized FDA's assessment. As part of assessment modernization effort, we created CASA, a knowledge management system meant to modernize the assessment of drug applications. CASA stands for Knowledge-Aimed Assessment and Structured Application. It is a data-based platform for structured quality assessments and applications that support knowledge management. We already have a fundamental knowledge base of the products, manufacturing processes, and facilities. And as new information comes to us in applications, we want to be able to assess that information in conjunction with our existing knowledge and really achieve knowledge management throughout the life cycle of the drug product we evaluate. While CASA is a key driver to fully achieve our vision of advancing quality assessment, powered by information technology and multidisciplinary approaches, we must integrate CASA with other key OPQ initiatives or programs. These initiatives and programs include Quality Surveillance Dashboard, QSD, Integrated Quality Assessment, IQA, M4Q, and Pharmaceutical Quality Chemistry, Manufacturing and Controls, PQCMC. And in next few slides, I will highlight what they are and explain how they relate or connect to CASA to provide a comprehensive approach enabling more effective and efficient quality assessment. CASA is a system that takes advantages of IT technology and innovation to modernize regulatory submission, assessment, and registration using structured data, advanced analytics, and knowledge management. CASA captures and manages knowledge, incorporates rules and algorithms for risk assessment, and enables assessors to perform advanced analytics, resulting in a comprehensive and science risk-based structured assessment. And to maximize the benefits of CASA, we need information and data that are well structured and organized. This is where ICH M4Q and PQCMC come into play. ICH M4Q is currently under revision. It will reflect advancements in technology and regulatory approaches, so it can continue to provide harmonized guidance on the content and organization of the quality information in an application across regulatory organizations. Then CASA will use information from M4Q for quality assessment to facilitate approval and lifecycle management and accelerate patients and consumer access to pharmaceuticals. For CASA to effectively use information from M4Q, we need PQCMC, which stands for Pharmaceutical Quality, Chemistry, Manufacturing and Controls. PQCMC is still under development. It provides a standard data elements and data exchange standards to the industry so that future submissions will contain structured quality information to be used by the CASA system. It is a critical enabler for M4Q implementation and long-term effective knowledge management. Therefore, to enable an efficient and effective quality assessment and fully take advantage of CASA, we need applications that incorporate both organization as defined by M4Q and data standards as defined by PQCMC. To assure seamless integration of all the relevant quality disciplines in assessment of drug applications using CASA, we have integrated quality assessment teams and processes. In this context, the assessment is done by a multidisciplinary team, followed a defined business process with clear roles and responsibilities. And this figure shows the relationship of IQA with respect to CASA, M4Q, and PQCMC. An assessment of a proposed product should leverage relevant information available about the product and how and where it will be made. Quality Surveillance Dashboard, QSD, will augment CASA and really allow us to use current and historical information about both the facilities and the applicant in one place. Together with information from the application, we will be able to conduct a comprehensive assessment of the application by considering all relevant risk factors. I want to emphasize that using these advanced tools or systems are expected to enable us to do our quality assessment more effectively and efficiently by applying the same quality standards and at the same time improve the consistency of quality assessment. Among these tools, CASA certainly plays an important role in assuring the quality and consistency of our assessment. In this context, some key features of CASA worthwhile highlighting here are captures and manages knowledge across the life cycle of a product, has built-in rules, algorithms, and analysis to inform regulatory decision-making, focuses on structured regulatory quality assessment, and provides data integration with other FDA systems that I just mentioned. OPQ is focused on continuing CASA's development. Our vision over the next 5 years includes expanding CASA to drug substances, including DMFs, new and generic drugs applications, all dosage forms for generic drugs, INDs, new drugs, and biologics applications, as well as post-approval changes or supplements. I would also like to point out that in the November 2022 Pharmaceutical Science and Clinical Pharmacology Advisory Committee meeting, we also gained unanimous support from the committee for expanding the use of CASA from generic drugs to new drugs and biologics. In conclusion, CASA and other quality assessment initiatives that I just presented are driving innovation in our quality assessment, powered by our modern technology in IT. I would like to thank our OPQ and CDA staff, as well as CDA leadership for their support of our modernization effort in quality assessment. And with this, I would like to conclude my presentation and thank for your attention. Thank you.
Good morning. My name is Susan Kirshner, and I am the Director of the Division of Biotechnology Review and Research 3 in the Office of Biotechnology Products and Cedars. And I'd like to thank the conference organizers for giving me the opportunity to discuss patient-focused specifications with you today. Before I launch into my talk, I'd like to give you a brief overview of what I will cover today. I'll begin with a discussion of patient-focused drug development as background, and then discuss specifications and provide some guiding thoughts about the selection of tests, analytical procedures, and acceptance criteria. And I'll close with a summary of what I've covered. Patient-focused specifications are a component of an end-to-end patient-focused drug development process. And that process includes patient-focused drug development, the manufacturing control strategy, and mature manufacturing quality systems. Patient-focused is about ensuring that patients' experiences, perspectives, needs, and priorities are meaningfully incorporated into decisions and activities related to their health and well-being. Patient-focused drug development is a systematic approach to help ensure that patients' experiences, perspectives, needs, and priorities are meaningfully incorporated into medical products throughout the medical product lifecycle. Patient-focused drug development helps ensure the acceptability and usability of the drug by the patient and promotes appropriate use of the drug. In 2012, FDA established the Patient-Focused Drug Development Initiative to more systematically obtain patient perspectives on specific diseases. I now want to begin focusing on specifications and their role in the manufacturing control strategy. The manufacturing control strategy should be designed to ensure the quality of each lot of drug product, and specifications are just one component of the overall control strategy. Specifications are standards that the product must conform to prior to lot release. And they're used to confirm the quality of products, intermediates, raw materials, reagents, components, in-process materials, container closure systems, and other materials used in product production. To develop patient-focused specifications, several things are needed. Firstly, the quality target product profile should be patient-focused in design to ensure that the product meets user needs. As such, the target product profile should ensure that the product is safe, pure, potent, and usable by the patients. Some examples of poorly designed products that we've encountered include subcutaneous injection volumes that are too large to be delivered into a single site, so multiple injections are required to deliver a full dose, pills that are too large for patients to easily swallow, and syringes and auto-injectors that are difficult for impaired patients to use. Development of patient-focused specifications also requires that product quality attributes are well characterized. Attributes that are critical to the patient experience with the drug should be identified, and their impact on the safety and effectiveness of the drug should be understood. Because biological products are mixtures of the desired product and process and product-related impurities, the impact of all these components on drug product safety and effectiveness should be considered. Lastly, to develop acceptance criteria for specifications that focus more heavily on what is important to the patient and less heavily on manufacturing history, the manufacturing facilities where drugs are made should have mature quality systems that focus on outcomes that affect the patient or consumer. So now let's start talking more deeply about specifications. Specifications have three components. The test, which is about what attribute we are testing. For example, purity, impurities, and potency. The analytical procedure or method that will be used to evaluate the attribute being tested. For example, HPLC and SEC are common methods used to test for both purity and impurities, whereas bioassays are frequently used to test for potency. And the acceptance criteria that are used to decide whether to accept or reject a lot or batch. Acceptance criteria generally include numerical limits or ranges, or sometimes other criteria such as positive or negative if a threshold method is being used. Acceptance criteria also include the sampling plan, which is a description of which samples are going to be tested. As I just said, specifications are comprised of the test, the method, and the acceptance criteria. In the next 4 slides, I will discuss test and method selection, and then spend about 8 slides discussing acceptance criteria, and close with a brief case study. Test selection is product specific and should confirm the quality of the product. Furthermore, tests should be selected based on the quality target product profile. It's kind of a mouthful. For example, it is generally not necessary to test antibody-dependent cellular cytotoxicity when the target is soluble rather than membrane-bound, because ADCC doesn't happen under those conditions. As another example, the mechanism of action for some enzyme replacement therapies require that the drug be taken up into cells. For those therapies, cellular uptake assays may be a release test. However, cellular uptake assays are not needed when the target of the drug is in the blood. Test selection should also be informed by understanding the product. For example, if there are data that trisulfide bonds reform to disulfide bonds in vivo, then it may not be necessary to control trisulfide bonds. Similarly, C-terminal lysines and monoclonal antibodies are generally known to not impact the structure and function of monoclonal antibody products, and release testing for C-terminal lysines is not requested. Another consideration for test selection is that critical quality attributes that can change during storage should be evaluated in stability programs. The selection of tests should also be informed by process understanding. That is, knowing what components you put into your manufacturing process, how those components get broken down, and whether your manufacturing process removes those components. For example, release testing of process-related impurities such as methotrexate, insulin, antifoam, host cell proteins, and host cell DNA may not be needed at release if removal of those impurities is validated and perhaps monitored by in-process testing. I also would like to note here that release testing of those same in-process impurities is generally needed under ND because the manufacturing process is still under development and removal isn't validated. Finally, release and stability testing of primary container closure systems to ensure functionality is also needed. For example, we ask for container closure integrity testing to ensure sterility on stability, and for prefilled syringes, we ask for break loose and glide force testing. This slide provides the generally expected release tests as noted in ICH Q6B, specifications, test procedures, and acceptance criteria for biotechnological and biological products. Some tests are also required by regulations. For example, sterility testing is required of parental products, and moisture testing is required for lyophilized products. Now that we've determined what attributes should be tested, we need to decide what analytical methods should be used. One consideration for method selection is the attribute being tested. For example, for sterility testing, you would use a microbiological method, whereas testing of purity and impurities, you would generally use methods that separate components of the mixture in a way that can be measured, or that individual components can be measured. In addition, some attributes may need to be evaluated using more than one method to be fully understood. In those situations, it is expected that the methods will be based on different principles, which we sometimes call orthogonal methods. So, for example, For impurities testing, it's common to use different methods so that size variance, charge variance, and hydrophobic variance can be evaluated. In addition, it's critical that the analytical procedure is shown to be suitable for its intended purpose, and if the attribute being tested changes on stability, then the method should be able to detect those changes. Now I want to shift focus and talk about some of the underlying principles for setting the acceptance criteria for specifications. Acceptance criteria are defined as the numerical limits, ranges, or other criteria for the test described. So the question is, how can we set patient-focused acceptance criteria? To address this, at least in part, I want to refer to the Manual of Policy and Procedures 5017.2 on establishing impurity acceptance criteria as part of specifications for NDAs, ANDAs, and BLAs based on clinical relevance. Also for this talk, I will primarily discuss acceptance criteria for products regulated under BLA. First of all, acceptance criteria are frequently set on a case-by-case or product-by-product basis because acceptance criteria may need to take into consideration the risk of an attribute to product safety and efficacy, the clinical experience with that attribute, and the context of use of the product, such as the dosage form, dosing regimen, route and duration of administration, clinical indication, and intended population. For example, we may allow for higher levels of endotoxin per product dose if that product is going to be administered IV over several hours as compared to being administered over a half hour. In general, the types of data and information provided should be guided by consideration of the clinical impact of impurity levels rather than manufacturing process capability. This will help ensure the acceptance criteria are clinically relevant. However, for biotechnology products, the relationship of impurities to stability, potency, and adverse clinical events may not be clear. And in such cases, we may place more weight on manufacturing process capability when setting acceptance criteria. In general, acceptance criteria should be supported by a risk assessment that includes information on the impact of the impurity on activity, pharmacokinetics and pharmacodynamics, safety, and immunogenicity. Data and information can come from a variety of sources, such as clinical, non-clinical, and analytical data, prior knowledge, and publicly available information. Also, it's important to understand that uncertainty about the attribute or quality of the data or knowledge can be a factor in assessing the strength of the risk assessment. Two examples of sources of uncertainty are the strength of the data to understand the clinical effect and the capability of the analytical method used to assess the attribute. We recommend that you apply risk management principle on managing uncertainty to your risk assessment. For example, principles that are described in ICH Q9, which recommends addressing uncertainty through the use of knowledge. In summary, to more fully implement patient-focused acceptance criteria, information is needed to bridge the gap between process capability and relevance to the patient. Okay, so now I want to end with a case study about setting acceptance criteria for a drug that is known to deamidate during storage. So, in the top bar in red is the clinical experience with levels of deamidation, which also corresponds to this black bar, which represents the manufacturing experience with that product during development. Stability studies indicated that deamidation will continue over time, and the expected change in deamidation over the proposed shelf life is shown here in green and here in red. In the license application, the applicant requested deamidation acceptance criteria as shown here in red, then added to that the expected change in deamidation over shelf life. And so this line in black represents the full range of DP end of shelf life acceptance criteria the sponsor requested. And the circled area represents the amount of the range of data or of information that was not shown in was beyond clinical and manufacturing experience. To support this request, the applicant provided information from in vivo and ex vivo studies to show that the product rapidly deamidates in blood and that potency is not impacted by deamidation. There was also knowledge that this attribute did not contribute significantly to product immunogenicity. The information provided justified the requested range, and the proposed acceptance criteria were accepted. So in summary, I'd like to reiterate that patient-focused specifications are a component of end-to-end patient-focused product development that includes patient-focused target quality product profile, product characterization, a mature manufacturing quality systems, and a manufacturing control strategy that is comprehensive. Patient-focused specification setting includes using the patient-focused target quality profile as a guide in selecting the appropriate tests and analytical procedures, and using product knowledge to support the acceptance criteria. However, uncertainty in setting acceptance criteria may arise when the relationship between an attribute and the impact to patients is not clear. Risk assessments can be used to address this uncertainty, but the risk assessment should be supported by data and information. Sources of data and information that can be used to address uncertainty include clinical, in vitro, in vivo, and ex vivo data, prior knowledge, and publicly available information. If uncertainty is not adequately addressed, there may be greater consideration for manufacturing process capability when setting acceptance criteria. So as a closing thought, I'd like to state that OPQ is committed to patient-focused drug development, and we encourage sponsors to incorporate end-to-end patient-focused product development information in their submissions. So thank you for your attention today.
Thank you for the great presentations. We'll now enter our 15-minute break. Please enjoy your break. Welcome back from the break. We hope you're able to refresh your coffee or tea to help move us right into the next presentation on modernizing quality assessment of new drugs by Dr. Hong Kai, who is the Division Director in the Office of New Drug Products, OPQ. And our final presentation in this session before the Q&A panel is on collaborating for generic drug development and approval success, a regulatory perspective, by Dr. Helen Tang, who is the Division Director in the Office of Life Cycle Drug Products, OPQ. Now, please join me to welcome Dr. Kai.
Good morning, good afternoon, and good evening. Welcome to 2023 Pharmaceutical Quality Symposium. I would like to thank Ray for the nice introduction. My name is Hong Kai, and I'm the Division Director of the Division of New Drug Product 2 in the Office of New Drug Product and the OPQ. My office is responsible for the quality assessment of the small molecule pre-marketing applications and the PDUFA program, such as INDs and NDAs. Today, I'm excited to be here to discuss our office initiatives in the effort of modernizing quality assessment. In my today's talk, first, I would like to present you the challenges we're facing with quality assessment of INDs and NDAs. Then I will introduce to you our recent development of CASA for new drug quality assessments. This includes CASA for INDs and the updated features in the CASA for drug substance system. CASA represents knowledge-aided assessment and the structured application. Then I will provide a summary of my talk. INDs and NDAs contain the drug development histories. Drug development is a complex and sometimes a long process. As illustrated in this diagram, the drug development from discovery to marketing involves multiple stages. An IND will be submitted to FDA when the sponsor is planning to use the investigational drug first time in human subjects. Subsequently, when new data is collected, there will be amendment, including meetings submitted to FDA. If the drug candidate shows promising marketing potential after going through clinical trials, the sponsor will submit an NDA for marketing application to FDA. Accordingly, the FDA's review of those regulatory submission activities is shown here. This process is a collaboration between the researcher, drug developer, and FDA. For the box highlighted here are the submissions and assessment activities under the respective IND. INDs mainly fall into two categories: commercial or sponsor investigators INDs. The purpose of commercial INDs are intended for marketing their proposed drug products. The sponsor investigator INDs, also referred as research INDs, are for research purpose. Most commonly, the sponsors for research INDs are physicians. There are also expanded access INDs. Both commercial sponsors and the sponsor investigators could submit such INDs. The purpose is for expanded access to an investigational drug for treatment or emergency use for their patients under certain conditions. This table shows the historical IND activities in CDER for the years between 2018 to 2022. It is listed in two columns: new INDs and INDs with activities or submissions. The new INDs include both commercial and research INDs submitted at that year. The INDs with activities include the submissions from both new and existing INDs at that year. This table does not include biosimilar biologic INDs, expanded access INDs, and unknown INDs as shown in the footnote. Expanded access INDs are listed in a separate table on the right. The most recently available 5 years data for expanded access IND is from 2016 to 2020. This data is available on the FDA website as shown here. It is noted that there are more than 10,000 INDs
with activities or submissions yearly. In general, the trend of the number of IND-related submissions or activities are on the rising. For new INDs, the total number submitted per year is still trending up overall since 2018, with some ups and downs. The highest number submitted in the year of 2020 during this 5-year period. There is a clear trend of increase of the number of the expanded access IND submissions from 2016 to 2020. Further, there are also approximately 200 NDAs per year for review. As you can imagine, there is a significant quality review task presented to the agency. When a sponsor submits an IND, it will come through Health Authority Gateway as a PDF document. The quality assessment document is also in PDF format. Those PDF documents are stored on Health Authority local servers. They are not easy to search. Additionally, those assessment documents could be lengthy, unstructured text narrative with critical information dispersed in many assessment documents for a single application. As a result, it is lacking efficient information sharing, knowledge management, and data analytics among those assessments. From submission point of view, INDs often have long histories. There are numerous types of INDs with various objectives. Many research INDs may not be submitted in eCTD formats. The technical complexity of the submission is increasing, such as novel dosage forms, emerging technologies. Most often, the review is under tight timelines in order to bring new drugs to the market for unmet medical needs. The number of submissions are increasing. Therefore, it brings review challenges with a large amount of information, but often dispersed in multiple places of an IND or NDA assessment. This resulted of difficult knowledge management, hampering holistic review, low efficiency, potentially redundant work, and inconsistencies. It hinders the effort to produce high-quality reviews for stakeholders, such as Congress, pharmaceutical industry, and most importantly, the patients. Therefore, we need to introduce 21st century technology to assist the quality assessment. In 2016, FDA's CASA system was envisioned as a means of modernizing FDA's assessment by utilizing structured data, advanced analytics, and knowledge management. CASA is the acronym for Knowledge-Aided Assessment and Structured Application. FDA's CASA system is a data-based platform for structured quality assessment and applications that supports knowledge management. Instead of a long narrative PDF document, the quality review will be performed using CASA system, which is stored in the cloud server. When fully developed and implemented, the information sharing will be in a standardized and structured format. CASA will enrich the effectiveness, efficiency, and consistency of regulatory quality oversight through lifecycle management of products and facilities. CASA system will significantly help the assessors to reduce the time spent for locating information, performing cut and paste, rather by using the time in the critical thinking and the regulatory decision-making. Ultimately, CASA will advance FDA's focus on pharmaceutical quality, the foundation for ensuring the safety and efficacy of drugs.
CASA for New Drug System is built on the success of CASA for generic, with added flexibility to meet various needs for new drug assessments. It facilitates risk-based assessment and promotes innovation, involves users in all stages of development, from prototype to production, including testing, implementation, and optimization. The chart below provides an update on the current status efforts and plan for future development of CASA for new drug. CASA for drug substance has been developed and completed. It has been implemented for the assessment of new and generic drugs since March 2023. CASA for IND, the internal prototypes, is under testing. In the coming year, we're planning to develop biopharmaceutics, manufacturing, and the new drug product modules for the NDA assessment. When CASA IND system was designed and built, CMC characteristics are taken into the considerations. CMC changes are quite common during IND stages. Meanwhile, some of the INDs share the common CMC information through letter of authorization provided by sponsors. For example, different INDs may share the same API. Same investigational drug product may be studied in multiple INDs for different objectives. INDs could also contain various developmental goals. Although many are for marketing purpose, but there are also some for research, expanded access and treatment, as shown in the tables of one of the previous slides. Therefore, CASA IND desired features are flexibility, enhancement of knowledge management, facilitate innovation. In order to ensure CASA system meets the various assessment needs for IND, we have designed the CASA IND system with the built-in flexibilities through the module and the block approach. CASA IND contains 3 modules: overview, drug substance, and the investigational drug product. In each module, there are numbers of blocks to capture the essential background and the CMC information assessments in CASA to ensure a high-quality review and knowledge management. The assessor can select the necessary modules and the blocks based on the content in the submission and the assessment needs. They can select all the available modules and blocks or some of the modules and the blocks. For example, if a sponsor has made a reference through a letter of authorization to allow FDA to review a DMF in support of the API requirement for their IND, the assessor can select Overview and the Investigational Drug Product modules. CASA will record and connect the drug substance assessment to the referenced DMF. The CASA IND system with the structured assessment data will enable the connections of the relevant assessment information from different applications. CASA IND also provides assessors the flexibility to select some of the blocks from each module for an assessment. In another example, when an assessor is reviewing an IND amendment that only contains updated CMC information related to the investigational drug product specification, batch data, analytical methods, as well as the stability data for both drug substance and drug product, CASA system will automatically populate the necessary IND background information from previous review. The assessor can focus their time and resource on the assessment of the CMC changes by only select those relevant blocks and modules as shown here. For any single IND, the submission contains the drug development history as shown in the diagram. Similarly, CASA will capture the entire quality assessment history for the same IND. Therefore, subsequent reviews can be automatically built on the previous one. It will refresh the assessors with regards to the application history, assists regulatory decision-making, and increase efficiency. Additionally, it will also benefit the workload management and the resource planning. CASA can automatically link those INDs who share the same CMC information through a letter of authorization as illustrated in the diagram. This will make it easy to manage and track INDs with shared CMC information through letter of authorization,
enable to access previous quality assessments for parent and daughter INDs easily, optimize assessment and knowledge management, and improve efficiency. Now I'm going to switch gears to introduce CASA for drug substance recently added enhancement. CASA for drug substance has been added the capability to utilize machine-readable structures, SD file for chemical structures. The assessment goal is to quickly identify potentially high-risk impurities, apply consistent standards for assessment of drug substance information in NDAs, ANDAs, and DMFs, inform decision-making, and increase efficiency of assessment. Chemical structure data file format that can associate data with one or more chemical structures. Tables of information can be translated into structures, which can then be searched. Errors are eliminated. Easier to transmit structures to other offices or stakeholders within the agency to facilitate multidisciplinary review and collaborations. The chemical structures and the synthetic pathways shown here are pictures or PDF files, and they are not machine readable. On the right side, those are machine-readable GSRS forms. Those structures can be read and searched by the computers. In a desired state, drug substance synthetic routes in CASA can be searched and analyzed. The analytic tools will enable CASA to search based on drug substance, reagents, solvents, impurities, and display synthetic pathways. The goal is to identify reaction chemicals that potentially generate high-risk impurities, track global supply chain, identify supply chain vulnerability. For example, if a drug substance is synthesized through 3 steps of reaction from the starting material, based on the internal build algorithm, the computer can search those structures to identify the high risk of genotoxic impurities, such as nitrosamine impurities, so the assessor can assess whether there is adequate CMC control to ensure the patient's safety. CASA system is a tool to modernize institutional knowledge management, built-in data analytics to improve assessment quality, improve assessment efficiency and consistency, inform decision-making, and facilitate innovation. Ultimately, it can help to bring more affordable and accessible medicines for American patients. In summary, CASA for new drugs are built using the same approach as CASA for generics, but include unique elements such as flexibility and analytical tools based on the needs of IND and NDA assessments. CASA for IND prototype is undergoing pilot testing to optimize its module for various assessment needs. CASA for drug substance interface has enhancement features which has been released in CEDAR IT platform. CASA for new drug enables knowledge management, increases the critical thinking time for the assessor, and promotes consistency and efficiency in regulatory assessments. Before I end my talk, I would like to thank OPQ CASA for IND working groups, OPQ CASA for drug substance working group, OPQ CASA for drug substance nexus implementation group, all developers, super users, testers, and staff implementing CASA, our host CEDAR Small Business and Industry Assistant team. And thank you for your attention.
Good morning, everyone. Today, I'm very excited to have this opportunity to discuss how collaboration between generic drug companies and regulatory authorities can accelerate the approval of generic drugs. I will begin by highlighting key challenges faced by both industry and regulatory authorities. By gaining a deeper understanding of each other's situation, we can work together more effectively. Then I will discuss the benefit of industry regulatory collaboration. Following that, I will touch on the agency's effort to strengthen this collaboration and what's expected from generic drug companies. At last, I will conclude by discussing how we can align our efforts to achieve our common goals. For generic industry, you are in a highly competitive environment that requires constant innovation in product formulation and manufacturing process to remain competitive. You must carefully evaluate intellectual property and mitigate risks of patent litigation. And you need to tightly control costs while accelerating product time to market. Now, let's take a look at the challenges FDA is working to address. We are working very hard to ensure that the quality of generic drugs remain top-notch as more and more generic products entering the market. We need to improve the efficiency of assessment with our limited resources. We need to work diligently to prevent and address drug shortages so that everyone has access to the medicines they need. At the meantime, we need to keep up with the fast pace of science and technology advancement. We have a big task ahead of us, but we are up to the challenges. Both of us are facing a lot of challenges. How can we successfully handle these challenges? Well, the answer is simple. That is collaboration. We had some great success stories that we worked together, like teaming up to fight the pandemic and programs like PEPFAR. When generic companies and regulatory authorities engage in true collaboration, we are able to calibrate expectations, be more transparent, and build trust. By keeping the lines of communication open, we can identify and solve problems early. When we take a collaborative problem-solving approach, I believe we can continue to overcome challenges together. Now let's take a closer look at what we are doing from a regulatory standpoint to boost our collaboration with the industry. First, we are committed to modernizing regulatory frameworks. Just as discussed in the previous talk, we are putting CASA, knowledge-aided assessment and structured application into action across the board, covering both generic and brand name drugs. CASA is designed to capture and manage knowledge during the life cycle of a drug product, establish rules and algorithms to facilitate risk identification, mitigation, and communication. For generic drugs, we have already rolled out CASA for oral solid dosage forms, and we are working on expanding its use to liquid dosage forms. We are also putting a strong emphasis on transparency. For example, we currently published the guidance for nitrosamine, drug substance-related impurities. The purpose is to keep it straightforward for all parties involved. Efficiency is a top priority for us, which is why we are currently cross-training all our review staff for different dosage forms. This ensures that we become more adaptable and agile in our operation. In terms of communication, we are actively creating more opportunities to engage with the industry. This allows us to gain a deeper understanding of the challenges you are encountering and make sure that we are all on the same page. There is no doubt that we have made great progress, but we see this as an ongoing journey. Our goal is to make our collaboration better and better. Constructive communication plays a vital role in fostering trust and ensuring consistency. Now let's explore the various pathways for pre-ANDA communication, including control correspondence, pre-ANDA product development meeting, also referred as PDEF meeting, ANDA pre-submission meetings, also referred as PSUB meetings. These channels provide valuable opportunities for us to connect. In the spirit of collaboration, we also have expectations from the generic industry. First, we expect the industry have a thorough understanding of all relevant guidance. This knowledge is very important for preparing a first-class ANDA pre-meeting package. We also expect any request to be based on solid data rather than unsupported claims. We expect generic companies to carefully select the most appropriate meeting format and communication channel for specific inquiries and ask clear, precise questions. By aligning our efforts, both generic companies and regulators can work together to streamline review timelines and expedite the availability of high-quality, affordable medicines. However, this work requires a mutual commitment from all parties involved. To submit a controlled correspondence, you should follow the draft guidance for industry, controlled correspondence related to generic drug development. You'll also need to check questions and answers on quality-related controlled correspondence first at this link. The most frequent controlled correspondence topics have been grouped into different categories and may save you some time. To receive the most accurate and fast response from the agency, it is important to be specific, concise, and clear in stating your inquiry. This clarity helps us to answer your question accurately. In addition, if your questions are multidisciplinary and require input from another sub-office, your control correspondence will be characterized as a level 2 instead of a level 1 control correspondence. This means the time frame for a response will be extended from 60 days to 120 days. To avoid delays, please consider separating your questions accordingly. So you may wonder, should I submit a control correspondence or request a PDEF meeting? For control correspondence, if your questions is just a single question or closely related questions, you should submit control correspondence. And also, if your questions are outside of PDAC meeting scope, then you should go with control correspondence. As I said before, control correspondence has two levels. Level 1 and level 2, they have different response time. And control correspondence is also good for post-PDAC meeting clarification. For PDAC meeting, it is best for multidisciplinary questions. And if you have new questions that are not addressed in control correspondence, generally PDAC meeting will be held within 120 days if granted. And remember, do not duplicate control correspondence in a PDAC meeting. Pre-ANDA meetings serve two main purposes. First, they help to clarify regulatory expectations from the get-go. Second, they work to improve assessment efficiency, potentially reduce assessment cycles, ultimately speeding up the approval process. It is important to note that about 30% of meeting requests face denial. The primary reasons for these denials are incomplete meeting packages, questions fall outside of the intended scope of the meeting, requests for pre-assessment, questions that do not contribute to clarification. Ensuring your meeting request addresses these factors can significantly improve your chance of a successful pre-ANDA meeting. A PDAC meeting request will be granted if they can meet the following criteria. It is a complex generic product. There is no product-specific guidance, PSG, available. When a PSG is available, but the applicant is proposing an alternative bioequivalence approach, the meeting package is complete, including all generated data and specific proposals for product development. This should include details about proposed development plan, such as an alternative study design, along with just sufficient justification to support a proposal. It is clear that controlled correspondence wouldn't be able to address the questions raised by the applicant adequately. As a suggestion to generic industry, we encourage you to have clear goals in mind when interacting with the agency. Ask specific questions about your program and approach. This will help us provide targeted and effective advice. Justify your proposal with preliminary data. However, no data dumping. And avoid assessment issue type questions. For example, do not ask about acceptance criteria for specification, acceptability of the study result. So this criteria just to ensure that PDAC meetings are granted in situations where they are most beneficial and necessary. When it comes to pre-ANDA meetings, it's important to know that there are two types of meeting, PDAC and PSUP. The choice between them might leave you wondering, and it is critical to get it right because submitting a meeting request in the wrong category would lead to a denial. So let's break it down. PDAP meetings are intended for scientific exchange on specific issues. This includes discussion of proposed study designs, alternative approaches, additional study expectations, or any questions you may have. At a PDAP meeting, the FDA provides specific advice on your ongoing and the development program. On the other hand, PSUB meetings provide an opportunity for ANDA applicants to submit unique or novel data or information to the FDA. This data will be included in the application material submissions. PSUB meetings are typically held 6 to 12 months before you plan to submit your application. Now let's go over the criteria for a PSUB meeting. We recommend for complex products, you should first seek FDA's input through a PDAP meeting before requesting a PSUB meeting. This ensures that the FDA is aware of the development program at the time of the PSUB meeting. However, you can still request a PSUB meeting whether or not you have had a prior PDAP meeting. FDA will grant a PSUB meeting under the following circumstances. If you had previously been granted a PDAP meeting for the same generic complex product. If the FDA determines that a PSUB meeting would enhance the efficiency of the ANDA assessment process. So, in short, collaboration is the key to mutual success. Utilize communication channels effectively. Make sure your proposal is focused and well-reasoned before submitting them. By following procedures, we can work more effectively to achieve our goals. That concludes my presentation. I will be back a little bit later on the panel to answer your questions. Let's join our hands to accelerate the delivery of high-quality generic products to the public. Finally, I would like to express my gratitude to my colleagues for their invaluable support. Thank you very much.
Thank you all for the great presentations. We're moving into our first Q&A panel, and as to our audience, if you haven't had a chance to submit your questions via the Q&A chat pod, please do so now. We'll wait a reminder for all of our panelists to please connect your mic and your video so that we'll be able to bring you up on screen. As a reminder, also to our audience, we'll answer as many questions as time allows. So we're waiting on our presenters to connect their mics and their videos. There may be just a slight delay as we move into this section of the question and answer panel. All right, we're getting some of our panelists coming on right now. We'll wait for everything to load very soon. And just for a mic check, our first few questions will be going out to Dr. Larry Lee. All right, we got another panelist coming on video there. And we've got Dr. Kirshner and Dr. Lee. Dr. Lee's loading. There we see Dr. Lee. And so while the other ones load, there's Dr. Kai is online. Awesome. And we'll just go ahead and move into our first few questions for Dr. Larry Lee. And here is the first question for Dr. Lee. Is the structured data and systematic approach to quality assessment a global initiative developed in collaboration with other regulatory agencies?
Yes, thank you for the question. That's an excellent question. I want to emphasize that, yes, I think the global harmonization in any quality areas are important to us. And then for this structural submission, yeah, we do plan to go through, have international collaboration. We do plan to go through ICH to actually look at, to focus on the so-called structured pharmaceutical submission. This is part of our future plan. So thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Lee. And here is the next question. Will CASA impact the standards for application approval from a CMC perspective?
No, I think I want to emphasize that CASA is really the internal system to FDA. I do not, basically, I don't think the industry will see any differences from externally. Simply because we are going to apply the same quality standards as well as apply the principles described in the published guidance. The CASA is really the system to help us to reduce to do our work internally more efficient or more consistently. So because of that, it will not have any impact on our review standard. And I also want to emphasize that ultimately, our assessor is still the one who make the final decision based on the information available in CASA or the information streamlined by CASA. Thank you.
Thank you, Dr. Lee. We do have one more question. It also involves CASA. And here is the question. What is the benefit of CASA for industry?
Again, I think the benefits from our perspective, as well as from more enterprise at the more enterprise level, is that CASA allows our assessor to do things more efficiently and consistently, so that when industry receive our comments on or our information requests, it will be more consistent. So I think our goal is that to really reduce variability in terms of our communication to industry, and the industry, therefore the industry will have a more predictability in terms of the question and the IR request from us. So I think that is definitely a benefit. And then also from our perspective, it will help us to tremendously to do our job more efficiently. Then we will continue to be able to meet our user fee commitments. Thank you.
Thank you, Dr. Lee, for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in for Dr. Susan Kirschner, and here is the first question for Dr. Kirschner. Does FDA accept cryo-EM method for large molecule identification, and does FDA keep up with the latest approaches of analytical methods?
Okay, thank you for the question. So I think there's two components to that. FDA does keep up with emerging technologies and the latest approaches, and I think Dr. Lee spoke a bit about our emerging technology program. And so if you do have an emerging technology that you're perhaps a technology that's been used under different circumstances and is now being applied in a different way, we do recommend you reach out to the Emerging Technology Program, and they can help shepherd the introduction of a technology into regulatory use. As far as cryo-EM specifically, I'm not sure I've actually encountered someone trying to use it that way. So in that sense, it might be an emerging technology. We are open to the introduction of new technologies. You would have to just provide the data in your submission to support its use the way or its intended use. I don't know, Larry, if put you on the spot, if you want to say anything more about emerging technologies, or I've said enough. All right.
Yeah, I think you're good. I think your response is good. Thank you, Susan.
All right, thank you both for responding to the right question. Moving on to the next question for Dr. Kirschner. And it's asking, Dr. Kirschner, from your perspective, in which phase of development should sponsors discuss their potential PFAC with OPQ?
Okay, so I'm assuming, thank you again for the question. I'm assuming PF there is patient-focused acceptance criteria. Typically you probably want to have that conversation somewhere in phase 3 or in later stage of development when you've got enough information about the product and your manufacturing process and stuff to support your acceptance criteria setting. But we don't, if you want to come in earlier with a question, you could certainly do that. But it seems like it would be most productive later in development. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Kirschner, and here's the next question. In the case mentioned, since potency is not affected by deamination, can the applicant put or use a wide acceptance limit?
Okay, thank you for that question. And I'm assuming we're talking about a wide acceptance limit for deamidation. And in the example I gave, we also, potency is not the only factor. Immunogenicity can be impacted by a deamidation, and sometimes PK can be affected by deamidation. So, you would need a comprehensive understanding of the variant or impurity to really to support your acceptance criteria. And overall, I would just say I'm going to kind of group, because there were a bunch of questions about setting acceptance criteria and additional examples, and I did give some. Basically we, with glycosylation, with various different components of the molecule, we have accepted acceptance criteria that were broader than perhaps manufacturing history or clinical experience when the appropriate information is available. And I think there's a fair amount of literature, particularly around monoclonal antibodies and the potential impact of different variants, deamidation, oxidation, glycosylation variants. You might want to look into that literature to see the kinds of information that people generate to understand how different variants and impact structure and function, and that those would be the kind of data that we would look to support acceptance criteria. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Kirschner, and it is the following. Can you please provide any additional examples of situations in which acceptance criteria beyond that shown in manufacturing process capability and clinical performance were accepted?
So like I said, and I think I brought a couple of these up during my speech. Glycosylation, variants, if they're shown not to impact, ADCC, or if the molecule doesn't have ADCC, then that would be an example. I think a lot of them do wind up being in the glycosylation range that we get a lot, sometimes oxidation. But like I said, you just have to understand the impact and justify the acceptance criteria, and then we'll be willing to talk to you about it. The other thing I want to bring up again is to reiterate that you have to have a well-controlled manufacturing process in order for us to be comfortable accepting broader acceptance criteria. So if your manufacturing process isn't well controlled and you don't have mature quality systems, then we may look a little more closely and be a little bit more conservative in our acceptance criteria setting. Thanks.
Thanks for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in for Dr. Hong Kai, and here's the first question for Dr. Kai. With the CASA desired state incorporating machine-readable structured data, is it logical to conclude that the future state should incorporate AI-assisted assessments?
Hi, that's a good question. And at this time, we have not utilized AI technology yet in our CASA IND development. But we will continue to explore and embrace new technologies for their potential use and assistance in our quality review to improve efficiency and allow our assessors to focus their time on critical thinking and resolving complex regulatory and technical issues for consistent and high quality review. So we are open-minded. Thank you.
Thank you, Dr. Kai, for responding to that question. We've got a couple more that did come in for you, and here's the next question. Will the eCTD structure still be applied when submitting information through CASA?
As Dr. Lee mentioned, the CASA system is mainly designed for our assessors to use to modernize our assessment as a tool to help improve efficiency and the consistency, as well as managing our knowledge. So at this time, I don't think there is a change yet for eCTD structure in terms of submission. But I would like Dr. Lee to add if there are any new trends and things and for this question. Thank you.
No, I don't think there's any changes. I think mainly, I think the folks should still follow the M4Q. And then I understand the ICH M4Q is currently under the revision. And then also in the near future, right, we will also focus on international collaboration or more like a structured data submission format, which will be used in conjunction to support the M4Q. I think those efforts will be in part will be going through ICH as well. So that's it. Thank you.
Thank you for responding to that question as a group. And we do have one more question for Dr. Kai. And here's the question. When do the NDA applicants need to provide SD files as part of the NDA submission? And is submitting SD files for chemical structures a requirement for CASA?
Hi, thank you for the question. We do encourage you to submit SD file as part of NDA submission, even at this time. However, please keep in mind, submitting chemical structure in SD file is voluntary. It's not a requirement. It is only one of the options to capture synthetic pathway and the impurities in a structured fashion. Even if there's no SD files, we are still capable to capture synthetic pathway and the impurities in a structured fashion. The key is we're trying to use a structured fashion of the submission, then we can use it for our assessment and analysis. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, Got a few questions that came in for Dr. Helen Tang, and here is the first question for Dr. Tang. After receiving a complete response letter for an ANDA, is it possible for industry to submit a controlled correspondence to FDA?
That's a good question. Yes, after receiving a complete response letter for an ANDA, the industry can submit control correspondence and just to seek clarification or address any outstanding concerns related to the application. Thank you.
Thank you for responding to that question. We do have a few more questions that came in for Dr. Tang, and here is the next question. Are there any restrictions or limitations on the number of controlled correspondences that an applicant can make?
That's a good question. The good news, no, there are not any restrictions or limitation on the number of times that the industry can submit control correspondence. As long as the submissions, the control correspondence are relevant, appropriate, and pertain to the application or regulatory guidelines. So the industry can submit controlled correspondence whenever necessary. Thank you.
Thank you for responding to that question. We've got another question, a couple more regarding controlled correspondences, and here's the next question. How long does it typically take to receive a response to a controlled correspondence?
That's another good question. The time it takes to receive a response to a control correspondence can range from a few weeks to 4 months, depending on the complexity of the question. So working the workload at FDA and our current priorities. So up to 60 days for level 1 control correspondence and 120 days for level 2 control correspondence. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Tang, and here is the next one. Is it possible to request meetings with FDA after submitting an application?
That's a good question. Yes, there are various meeting types that can be requested with the FDA. For example, the enhanced middle cycle review meeting and post-CRL scientific meeting, etc. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Tang in this round, and here is the question. It's regarding quality issues, quality-related issues. Do you believe that pre-submission development meetings are more effective than controlled correspondences?
That's a good question. Well, it depends. PDAP meeting allows for direct and real-time interaction with the FDA, facilitating in-depth discussion and immediate feedback. Controlled correspondence, on the other hand, can provide a written communication and allowing for specific questions and clarification. The choice between these two really depends on the nature of the issues and the available resources. Thank you.
Thank you for responding to that group of questions. Up to the beginning of our panel, we've got another question came in for Dr. Larry Lee. And here's the question for Dr. Lee. What is the future of CMC? And how is the industry changing? And also, what are the biggest challenges that we'll face?
That's an excellent question. I think the biggest challenges we are going to face in the CMC area is the volume of the submission, especially post-approval supplement will continue to increase, right? I think the speed and also the speed for drug development including the pharmaceutical development in pharmaceutical quality area, will continue to be shortened because we want to really make the drug available as soon as possible to the patients. So in that sense, I think the challenges is that how do we actually ensure we do a very effective work in a shortened amount of time, but the resources is still remain to be the same. So what we are trying to do here in the near future or in the future, our assessment will continue to focus on continuous improvement and to really utilize to take advantage of the IT or the advanced technologies to really help us to streamline, to make sure our assessment is effective as well as efficient as possible. So I think that is really what we'll focus on. And then CASA definitely is one of the solutions we are looking at, but we also continue to look for other ways to do it. But ultimately, I still want to emphasize that no matter what technology we use, our assessors still will make the final decision, and we will continue to train our assessor to make sure they have an up-to-date scientific knowledge, so they will be able to apply the risk-based principle to make the appropriate decision to support the approval of important drugs for the United States. Thank you.
Thank you for responding to that question. We have another question just came in from Dr. Lee and Dr. Larry Lee. And here's a question that's referring to one of the slides mentioned submissions going to a cloud environment. What is the long-term vision for submissions to FDA? Will the ECTD software and ESG be replaced by sponsors submitting to clouds shared by regulatory authorities?
Yeah, I think Hong Kong, feel free also to comment on it. I think this is also in your presentation slide. I mean, that is definitely one of our vision. Like, in fact, I think industry really want this to happen. I think as far as I'm concerned, and based on the conversation I had with the industry, we certainly support this vision. But we will do it collaboratively with industry to see what is the best way to approach it. Hong Kai, if you have any comment, please feel free to share.
Hi, yeah. Dr. Lee has summarized it well. This is definitely under the discussion, and we have been exploring different ways. But in general, the cloud environment is much more secure. It will be much more versatile. So as Dr. Lee elaborated, it's a collaboration between industry and the agency. Thank you for the question.
Thank you for responding to that question. Moving on to our next panelist, we've got a couple questions that came in from Dr. Susan Kirchner. And here's the next question. For some of the legacy compendial products, the test methods are with traditional techniques which are not able to fulfill current FDA expectations. In that case, if the application holder is going for an update of the new techniques, some challenges are faced to respond to the FDA queries. How can we deal with such cases?
Hi, thanks for that question. And Hong and you may need to, or Helen may need to chime in on this one from the small molecule perspective, because my expertise is in biologics. It is not uncommon to have methods updated either during development or during the product's life cycle. And so we're pretty accustomed to working through that. It usually requires some robust demonstration that the new method and the old method or a comparability between the old and new methods. It's not uncommon for the new method, particularly with no purity methods, to start seeing new impurities that were not seen before. That doesn't mean they were never in the product. It just means, generally the new methods are more sensitive. And the best way to deal with that is to have historic product on hand, or to make some force degradation studies and show, that these are impurities that were always there. And then we can talk about the extent to which anything new that we see needs to be controlled. But it's not an uncommon thing. And if you're having difficulties, you're welcome to come in for a meeting. Hong or Helen, do you want to chime in on the small molecules?
Hi, Helen. I can go first. Yes. So actually, Susan covered really well. And we do acknowledge the technological advancement, and there are some legacy method. So the key is you need to demonstrate the comparability of your new method is at least equivalent or better than the legacy method. And certainly you should, for any particular method and related to any specification, we do encourage you come in to discuss with the review team for each of them and to make sure it meets the intended purpose. Helen, you want to add something on that? Thank you.
Well, thank you, Susan and Kai. Actually, Hong, you have already covered pretty well, but for generic product, I just want to say, as long as you can validate your method, even though it's not USP method, we always have no issue with that. So don't worry about using any advanced technologies. We actually encourage that you can demonstrate you have a better method. We would like to see it. Thank you.
Thank you all for responding to that question. We've got a few more questions that came in for Dr. Kirchner. And here's the next question. How does quality systems maturity impact patient-focused specifications?
So, as I said earlier, if you want to be able to have specifications that are not based on manufacturing process capability, then you need a robust quality system that we're confident that, if you're seeing out of trend results, even if something is in specifications, you're going to go in and investigate what's going on with your manufacturing system. And so those are components of a mature quality system. Statistical process control is a great tool that should really be used to monitor quality system, both process performance. And, we would really like to have the focus be on the manufacturing process controls being a routine component of the manufacturing process, and not through endpoint testing. And if we can get to that place where you have a robust quality system and a robust manufacturing process, then we don't have to rely so much on end product point testing to demonstrate that the process is working. We can be more patient focused. Thanks.
Thank you for responding to that question. We do have one question. Another one just came in for Dr. Kirchner, and here is the question. How are acceptance criteria set if the impact of an impurity is not known?
Right, and I would really refer you to the map, I think it's 5017.2, because it does have some in-depth discussion about that. And, the bottom line is, unfortunately, if you don't have good information about the impurity, you are likely to be limited to your experience with that impurity or close to that. The more you know about the impurity, if you know what it is or what levels it is, if you understand its toxicity, then we have something to build off wider specifications. But if you don't know anything about it, then it's very hard to justify broader acceptance criteria. Thanks.
Thank you for responding to that question. Moving on to our next panelist, got a couple questions we'll try to squeeze in the very end for Dr. Hong Kai, and here is the first question. In addition to CASA for INDs, is there any plan to develop CASA for NDA quality assessments?
Thank you for the question. Yes, CASA for drug substance, as I mentioned, has been implemented in both ANDA and NDA application quality assessments. It is also our plan to develop other modules such as biopharmaceutics, manufacturing, and the drug product assessment for NDA assessment in the future. Similarly, as we develop the CASA for INDs, we will adapt existing CASA interface used for generics with the consideration of the needs of NDA assessments in design and the development of those modules for NDA assessments. Thank you.
Thank you for responding to that question. We've got another question for Dr. Kai, and here's the question. Can CASA INDs be used for different types of IND submissions, such as research, commercial, or expanded access?
Thank you for the question. So CASA for IND is still under prototype development and testing. And we're really working hard trying to optimize and make improvements. When it is fully developed under the production stage in the future, it is our intent for CASA IND tool to fit into the quality assessment needs of various IND types, such as commercial INDs, research, as well as expanded INDs. So the CMC characteristics during IND stages will be taken into consideration when designing CASA IND to ensure its flexibility, as well as meet the desired goal of enhancing knowledge management. Thank you.
Thank you for responding to that question. Another question on CASA for Dr. Kai is the following. Does CASA IND use artificial intelligence technologies?
At this time, no, we have not utilized that yet, but we are open for all kind of new technologies. So in the future, everything is possible, but we would make sure, as Dr. Lee mentioned, it's still the human power. It's the assessor making the decision, not the machine. It's our qualified assessment team put all the data together to make the holistic view and based on the totality of the data to make the risk benefit and the science-driven decisions. Thank you.
Thank you for responding to that question. We're going to bring in Dr. Kirshner back in to see if she can comment on CASA for BLAs.
Right, so I just wanted to mention that there are CASA modules that already have been launched internally for reviewing, in the CASA format, and we're developing additional modules for unit operations, and we're very excited about that. And so, the CASA program is expanded both to small molecules and large molecules, and we're very excited. Thanks.
Well, that's all the time we have for questions in our Q&A panel. And a huge thank you to our presenters and panelists for answering numerous questions that came in and being live there on camera. This takes us into our lunch break until 1 p.m. Eastern. Please enjoy your lunch. Welcome back from the break. From the lunch break, before we begin our next session, we'd like to remind attendees of a couple of housekeeping reminders. In addition to the speaker evaluation, we have our final evaluation. The final survey is the mechanism by which you will receive the certificate of attendance, which will support CE credits for RAP, SOCRA, SQA, and ACRP. When an attendee completes a survey evaluation, they can then download a certificate of attendance. The certificate must be downloaded immediately after completing the survey. Otherwise, this opportunity is lost. To simplify things, SBA will send our thank you email to all attendees, to all Adobe attendees tomorrow post-event, and that email will also have the survey link should you miss it over the next 2 days. Moving right into our first presentation in this session on inspections and a post-pandemic world by Alonzo Cruz, the Director of the Office of Pharmaceutical Quality Operations in the Office of Regulatory Affairs. And our final presentation before the Q&A session is on pre-license inspections, what industry should know, by Dr. Christopher Downey, Supervisory Chemist in the Office of Pharmaceutical Manufacturing Assessment, OPQ. Now, please join me to welcome our first presenter, Alonzo Cruz.
Hello, my name is Alonzo Cruz, and today I will be addressing some of the inspectional activities that we have been working on in this post-pandemic world. But before I jump into that, let me first explain the reorganization that ORA is going through. The new ORA model significantly impacts the human and animal foods program. However, there is still a significant impact to ORA's pharmaceutical drug program and the other medical product programs to be complete, in that our compliance functions and personnel will be transferring to our respective program centers. For the human and animal drug program, that will be to the Center for Drug Evaluation and Research and to the Center for Veterinary Medicines. Aligning inspectional resources with the centers and program priorities, our inspectorate offices and staffs will carry out inspections and other field-related activities for their individual portfolios. The Office of Pharmaceutical Quality Operations will be renamed as the Office of Human and Animal Drug Inspectorate. Go on and scan the QR code you see on the screen to get additional details regarding ORA's transition directly from FDA's website. Let me give a little history, or rather a timeline, on some of the activities that we have done since the onset of this public health emergency. On March 13, 2020, FDA initially suspended all on-site inspections. Those inspections that we continue to perform were those that were deemed mission critical to the agency. And early on, they dealt a lot with COVID-related activities, whether it was through inspections for respirators or for therapies associated with COVID-19. Routine foreign inspection trips resumed for us back in April 2022. We resumed trips to China earlier this year in May 2023. And throughout the entirety of the public health emergency, we continue to use alternative tools such as remote regulatory assessments. Were used when we could not get to an in-person inspection because of travel restrictions, or it was just not deemed mission critical for that in-person activity. Let me now take some time to talk a little bit about some of the work we did throughout. Some of the inspectional related activities that continued included our work with PICS, bilateral information sharing, and records requests in advance or in lieu of inspections. Also, during the time of the public health emergency, we developed a remote interactive evaluation tool that allowed us to, in addition to using tools of records request, work with facilities to have an in-video spotlight of manufacturing operations to assist in making those decisions the agency needed to make. We have and continue to be actively engaged with the Pharmaceutical Inspectorate Cooperation Scheme, PICS. This worldwide membership of some 54 participating medicines authorities includes well over 2,000 inspectors. This has allowed us to continue our work with our foreign regulatory counterparts, not only to work with them in developing harmonized standards and inspectorate tools and procedures, but also in the interpretation of GMP standard. This is critically important to help us strategically build not only collective trust, but to build capacity for the inspectorates really on a global scale. ORA also supported and supports bilateral information sharing with our foreign counterparts to gain a more universal understanding of the activities and the regulatory oversight that these countries pursue. There is no doubt that inspections are critical to FDA's oversight of pharmaceutical products. Though I should note that it's important that inspections are just one part of a robust, multi-pronged approach to overseeing safety and quality of FDA-regulated products using some of these various tools. Now, looking specifically at remote regulatory assessment tool, the agency uses RRAs when we determine that this tool would be an appropriate step in assessing compliance with FDA regulations. Through RRAs, we have and continue to assist us in assessing compliance with manufacturing processes, possibly to enable a firm to make corrective actions prior to our next physical inspection, to assess the adequacy of corrective actions taken in response to either previous inspections of complaints, which may lend to an increased time between FDA inspections, and certainly the use of an RRA may allow us to do that. If appropriate, to make decisions related to applications submitted to the agency without the need to conduct a pre-approval or other inspection. Now, we will continue to use these alternative tools to supplement physical inspections and the review of drug applications to assist the agency in making timely decisions on the status of facilities and the various products that they manufacture. Let me speak now for a few minutes to the work that we have done and have expanded within the mutual recognition agreement space. In 2017, FDA entered into a mutual recognition agreement with the EU. By mid-2019, all of the EU member states were assessed and found capable in their CGMP inspection portfolios. Since 2018, FDA has been requesting, reviewing, and classifying inspection reports from our MRA partners for sites assigned within our site selection model. In 2021, the U.S. entered into an individual MRA with the UK following their exit from the European Union. Of importance to note, during the public health emergency, we were really thinking creatively about how can we expand our partner network and the work that they're doing to help us in making some of the decisions that the agency had to make. So in 2021, a third country inspection assessment was done to expand the scope of MRA beyond just the United States and the EU. Essentially, it allowed the inspections that the EU member states have done outside of the EU to provide those inspections to the agency. It did greatly enhance, open up, if you will, more inspectional information that we were able to gather, and those inspections that we did outside of the U.S. to share with our EU member partners. We have now classified those inspections conducted by our MRA partners in third countries. Again, these are inspections that are conducted outside of the EU by EU member partners. Continuing the work that we have done in the MRA space, in July of 2023, the MRA, we now have a mutual recognition agreement with Swissmedic. And continuing expanding the work within the mutual recognition framework, over the past few months, we have taken numerous steps in preparing for expanding the MRA yet again. When the MRA was first implemented, it included pharmaceutical drugs for humans. The MRA now includes veterinary medicines as part of the portfolio within the MRA with the EU. There are about 16 EU member countries that are capable of the veterinary products and veterinary drugs as part of the MRA EU, and is greatly enhancing our ability to expand our network of inspectional activity within the pharmaceutical landscape. As you can see on this chart, it provides from FY18 to FY22 the MRA inspections that we have classified by fiscal year. You will note the red showing up in FY21. That is when we began in earnest of reviewing third country MRA reports. That number did go down slightly in FY22 as we began increasing the number of foreign inspections that we have been doing at FDA over that time. But our uses of third country MRA will continue in this post-pandemic world. Okay, so what can industry do? Certainly, I think ensure timely responses to the agency's request will greatly assist the work that we're doing. Ensure that the information that is submitted to us is accurate, complete, and updated, including registrations, submissions such as the Form 356H. Critically important. If there is, or if a company becomes aware of a disruption in either a supply chain or a supply of drugs produced at your facility, please reach out and contact the drug shortages staff immediately. They will help assist and navigate you through whatever regulatory challenges you may be facing as a result of the supply chain disruptions. And certainly, when utilizing contract manufacturers, have defined and established and documented activities to highlight and to demonstrate and show to them that you're taking GMP and compliance very seriously. It'll be important and will lead to fewer headaches down the line. Some final thoughts. ORA continues to serve as the agency's front line to ensure safety and effectiveness of products. We have resumed all foreign routine inspection travel, and that is important because we know that there is a significant amount of work to do, not only in the domestic space for human and animal drugs, but on that foreign space as well. ORA is making some transformative changes to enhance not only its efficiency and effectiveness, but we are continuing our enterprise-wide organizational changes to support program centers focused on inspectional activities. Thank you.
Good afternoon and happy Halloween, everybody. My name is Chris Downey, and I'm the Director of the Division of Biotechnology Manufacturing in CDER's Office of Pharmaceutical Quality. And today I'm going to give an overview of the pre-license inspection process for biologics, and hopefully I can convey some of the unique considerations for these inspections and provide you with some examples of challenges we have encountered along the way. So one area that we see inexperienced and smaller companies that are developing biologics run into problems is that they treat CMC development the same way that they might treat small molecule products or generics development. But biologics are not just big small molecules. The products are more complex and more heterogeneous, and their critical quality attributes are highly dependent on the manufacturing process. And many of the process parameters and controls are based on small-scale studies. And because of these complexities, process characterization, process validation, and process control are critical, and they also make pre-license inspections more complicated than many smaller or newer firms might be expecting if their experience is more in the small molecule realm. So here's just a quick overview of an example therapeutic protein manufacturing process going from the expression system through drug substance and through a final sterile drug product. And I'm doing this just to highlight in one handy visual reference for you the various ICH guidelines that apply to the different operations that our reviewers and inspectors rely on. So with this complexity for biologics come differing regulations and different expectations than from small molecules. And the first thing that I want to highlight here, because I think it can be a cause of problems on pre-license inspections, is that unlike small molecules where process performance qualification must be complete before commercial distribution, but may not necessarily be complete when the NDA or ANDA is submitted, for a large molecule, these validation activities must be completed when the BLA is submitted and the data are included in the application. And this means that inspectors on a pre-license inspection expect to see the final, what will soon be, hopefully if it's approved, commercial process and not a process in development. And I think this is a disconnect sometimes where inexperienced firms run into challenges because they're still getting their commercial process ready, but the inspectors are there and they're expecting to see the process ready to go and being performed as it will be performed after approval. Another area I want to highlight is that biologic processes rely on information that is sometimes overlooked, or at least let's call it underlooked, by applicants and facilities as they're preparing for inspections. And this includes small-scale data demonstrating things like viral clearance, shipping validation data, and protocols for reuse of materials like chromatography resins. Inspectors also evaluate risk from other products that are manufactured on shared equipment. For example, I've seen cases where a firm could receive an observation if the product that's under investigation is expressed in mammalian cells and is manufactured on shared equipment with a different product that's also expressed from mammalian cells, and that other product testing and controls do not conform to the virus control testing described in ICH Q5A. In other words, they're not testing for adventitious viruses in process. So even if the subject of the BLA product is conforming to ICH Q5A and has the expected testing, because it's on shared equipment with the non-conforming product, due to the increased risk of cross-contamination, there could still be a citation as part of the PLI. So when preparing for inspections, companies need to take this kind of supporting information into account and understand that it may be examined on the inspection in addition to the manufacturing operations and QC lab activities that are described in the PLA application. All right, so to step back for a second, you might be wondering why we do pre-license inspections. Why isn't it enough to simply certify that the facility has been evaluated in the past and is GMP compliant? And I think some of that is sort of self-evident from the unique considerations we've been discussing, but it's also true that PLIs are a statutory and regulatory requirement. So for your reference, I've listed some of the applicable rules and regulations around why we conduct PLIs here so that they're available if people want to look at them. Some key items to consider when preparing to submit a BLA include that all facilities in the application need to be registered with the FDA. Additionally, when submitting the BLA, you should provide a preliminary manufacturing schedule for both the drug substance and the drug product. And bear in mind when submitting that the facilities will need to be manufacturing the product during the BLA review cycle. It doesn't do any good if it's manufactured afterwards and we can't conduct a PLI. It's also important to understand that type 2 DMFs are typically not allowed for BLAs other than, for example, the small molecule component of an antibody drug conjugate. The BLA process is expected to be described in the BLA, and the applicant is expected to have knowledge and control over all aspects. And finally, I want to highlight, because I get questions about this a lot, how are biosimilar inspections different than other biologics inspections? And really the answer is they're not. Biosimilars are subject to the same rules and regulations and the same pre-license inspections as originator BLAs. So because of the distinct requirements and risks with biologics, it's important to consider how PLIs are distinct from cGMP surveillance inspections. I think the most basic difference is that a PLI is a product-specific and process-specific evaluation, and a PLI observes the application product as it is being manufactured. Another important difference is that PLIs are led by CDER staff who are biotechnology reviewers. In other words, the inspection staff come from the same divisions that are reviewing the CMC modules of the application, and they are specialists in biotech, and they are knowledgeable of the specific items in the application. And finally, a PLI is linked to the application review process. And this means that acceptable outcomes for both the application assessment and the inspections are required for approval. And this is true even if facility observations do not lead to a formal compliance action from the agency. If a facility is found not to be ready to support commercial manufacture of the specific product that we are evaluating in PLI, the application may still not be approved, even if there's not a formal warning letter or something like that. So diving in a little bit on how pre-license inspections work, PLIs use a systems-based approach to evaluate GMP readiness for BLA manufacture, and that covers 6 systems. Those systems are the product quality system of the establishment, the facilities and the equipment, the control and handling of raw materials, naturally the production system, packaging and labeling if applicable, and the laboratory control system. The PLA also evaluates the conformance of the process and the associated controls and supporting data with those in the application. You'd be surprised how often we see that the firm is doing something differently than what is listed in the BLA, or that there are whole processing steps being used that are absent from the application. So it's important to get on the same page between the facility and the applicant. And finally, there's an evaluation of data integrity and security. The FDA must be able to rely on the data generated at a facility and to know that those data are auditable and traceable. And it's important to understand that this includes not only the production data or the QC lab data, but also the supportive information we've discussed earlier, things like data that go into the virus clearance study. Some additional key items to consider for PLIs. As we'll see in the upcoming slides, the best thing you can do to avoid problems on inspections is to have a robust quality system in place. And this means that things like deviations, out of specification, or other nonconformances are promptly investigated and root causes are identified and corrective actions taken when needed. Additionally, a good quality system proactively identifies problems and takes corrective actions. It does not merely react to and justify problems. So let me give you an example. Hypothetical, but let's say it's inspired by real life events. Let's say we have one firm that detects a bioburden excursion, but they promptly identify the organism, they identify the source of ingress, correct that problem, look for other similar potential sources of contamination elsewhere in the process, and they also look at other processes in their facilities that might have similar risks. And then subsequent batches made thereafter no longer have the problem. And you compare that to another firm that has a bioburden excursion, and they basically rationalize that even though they had a CFU level beyond their control limit, the measured value is probably too low, it'd be a big risk to patients, and there's filtration steps later in the process that'll probably clear it. So they think they can release the batch, but they take no further actions. And then as we're looking, we see additional excursions on later batches. Which of these facilities do you think is going to have a better outcome on the inspection? Or for that matter, which of these is going to have fewer other kinds of problems? So this leads to another theme, which is we're investigating that the process is under control. For example, are the procedures and equipment in place sufficient to prevent contamination, or is the firm relying on detectability alone? And finally, it's important to understand that for biologics, the applicant must have knowledge and control over the process and the product. So we may ask to evaluate quality agreements between facilities and applicants to make sure that the applicant is really in control of the process. Okay, so shifting gears a bit, to illustrate the importance of inspection readiness in biologics development, here is the result of a recent informal survey of deficiencies listed in complete response letters. So this was looking back over a 3-year period and covered 32 total BLAs, including 15 biosimilars. An application may have more than one deficiency and may have been subject to multiple evaluation cycles, so the same BLA may fall into multiple buckets here. Note that the most common deficiencies were facility-related, and non-facility-related CMC issues were actually just as common as those related to clinical or clinical-related information. In addition, for biosimilars, facility deficiencies were more common than those related to demonstrating biosimilarity to the reference product. And I think this highlights that CMC development and selecting and preparing facilities to be ready for commercial manufacture are not areas to neglect or to put on the back burner when developing biologics. All right, so unpacking the inspection-related CR items a little further, we surveyed the underlying 43 observations and categorized them as well. And once again, there can be multiple issues for one application, and things can fall into more than one bucket. So as you can see, the most common issues are under the general category of quality oversight. Things like the firm is not consistently or sufficiently investigating deviations or out of specifications, or those investigations are untimely, things like that. In addition, they may have insufficient change management procedures. It's another common problem with quality oversight. Other commonly occurring issues are related to procedures and controls for low bioburden and aseptic processing. And again, I think this highlights the importance of being ready for inspection when the BLA is submitted. If a company is relying on FDA to find problems on the PLI and then take corrective actions during the cycle, you have to understand that those corrective actions often take time to implement and need data to support their effectiveness, and often result in an additional inspection. And the additional inspection would have to occur during the next review cycle. So all of these things can pose a risk to a first cycle approval, and it's better to be ready for prime time when the BLA is submitted. All right, so in summary, successful development of biologics includes ensuring that the manufacturing facilities are ready for commercial production and that they are ready for commercial production when the BLA is submitted. For biologics, facility and inspection-related issues have actually been the most common reason for CR in recent years. So again, it's not an area to neglect. Common biologic inspection issues include aseptic procedures, contamination, or cross-contamination controls, and environmental monitoring. And again, a recurring theme was insufficient quality oversight, the lack of thoroughness or timeliness of investigations and corrective actions. So just for reference, I've included a list of guidance and other resources that we rely on in preparing for and carrying out inspections. One thing I wanted to highlight is, even though we don't formally enforce the EU GMP Annex, I think it's current and has a lot of useful information regarding expectations for manufacture of sterile products and aseptic processing. So I think companies who conform with that guidance are better positioned for pre-license inspections, even though we don't formally enforce it. So with that, I'd like to thank you for your time and move on to questions. And again, happy Halloween, everybody.
Thank you for the great presentations. We'll give our panelists a moment to attach their mics and activate their video option. And as a reminder, as we move into the Q&A panel, if you haven't had a chance to submit your questions into the Q&A chat pod, please do so now. As a reminder, please remember to indicate the speaker to whom the question is being addressed, and we'll answer as many questions as time allows. All right, looks like we have one panelist up on camera. And we'll give just a moment for our other panelists to arrive on camera. All right, there we are. And we have both panelists on camera, and we'll direct the first set of questions to Mr. Alonzo Cruz. And here is the first question. Which one is more efficient, is a more efficient facility inspection? In-person or remote? And moving forward, will FDA utilize more remote inspections?
Okay, thank you for that question. And it's one that we have asked ourselves internally quite often in terms of which is more effective, a remote assessment or an in-person facility. In-person facility inspection. And I think each time we come up with the in-person facility inspection as the more efficient activity for the intended purpose of that of the inspection. With regards to continuing to use remote regulatory assessments as a tool, that will continue. One thing I want to draw attention to is that FDA had the authority prior to the public health emergency to use remote regulatory assessments or the records request as far back as 2012. So it's something the pandemic helped us to certainly utilize at a higher degree, but it's something that we will continue to use moving forward to help us when we can't get to a physical location for any number of reasons. Thank you for the question.
Thank you for responding. We have a few more questions for Alonzo Cruz, and here is the next question. Is there any work happening for MRA with Health Canada?
Canada and Mexico are two of our largest trading partners. We do have both bi and trilateral meetings with both countries, and we have active exchange of information, inspectional, health, and others back and forth. Outside of my office, there are evaluation as to whether or not we extend mutual recognition agreements, not only to Canada, but other countries potentially as well. So that work is continuing. But again, we maintain a very healthy exchange of information and relationships with Health Canada.
Thank you for responding to that question. We do have one final question in this round for Alonzo Cruz. It's kind of long, so we can repeat it if you need to. And here is the question. What happens when different regulators classify the same inspection differently? And also, what are the downstream implications of those different conclusions? And a final 3-question in one, this third part of this is, will it affect the applications in the US but not in the EU?
Okay, thank you for that question. Very intriguing. Let me start off by saying that when we are conducting an inspection in the US, or for that matter, when we are conducting an inspection overseas, we are using our compliance programs and filtering that through the laws through which FDA governs itself. So we're making that assessment not based on the foreign governments or foreign countries' rules and regulations, but those for approvability or compliance with FD&C, with the FD&C Act or other relevant guidelines. So I think the second part of that question is, probably the first question asked was, how do we react when we classify at the same site a foreign regulator and a U.S. regulator reaches different conclusions as a result of the inspection? And again, it goes back to what I mentioned before. We are both analyzing and filtering with the applicable laws relative to our country or our regulatory body. And there may be instances in which there are differences. A general GMP inspection at FDA is systems based, and the systems we may be doing an inspection on may be different from what a foreign regulator is, but there are any number of reasons why an ultimate final classification might be different. Again, we're basing those classifications on our respective regulatory frameworks. And I think there was a third part to the question, if you don't mind repeating it.
Certainly be happy to. And the last part is, will it affect the applications in the US but not in the EU?
Okay, so again, we are using, going back to now a strictly MRA perspective, in which we did not conduct an inspection, yet it is a site that has an application pending before us. And we would be looking at and evaluating their inspection report in terms of what they found and what factors they considered significant. And the FDA would make, again, its decision based on the regulations promulgated by the FDA. And those would be some of the factors that we would use in determining approvability of that particular application. Again, an inspection is just one aspect of the approvability process. And thank you for the question.
Thank you for responding to those questions. Moving on to our next panelist, we've got a few questions that came in for Dr. Christopher Downey, and here's the first question. They're asking for further clarification on inspection finding of facilities. Is this a poor design of aseptic areas or insufficient maintenance of process areas? Can you shed some light on that?
Yes, thank you for the question. The short answer is both. It pertains to, we certainly see issues with the poor design of aseptic areas, but we've also seen issues with utilities, for example, with water sourcing or water purification and things like that. So it's both.
Thank you for responding to that question. All right. we're moving on to the next one. We've got one that's addressed to Alonzo and Chris. So we'll bring in Dr. Downey first, and then you can pass over to Alonzo to see if he has any continuing thoughts on the following question. Given the complexity of large molecule processes, would remote inspections ever be considered for PLIs?
Yes, we consider remote inspections for PLIs. During the COVID pandemic, that was a tool that we were able to use in certain cases and learned a lot from. So as part of our decision making now, we do consider facility risk based on for example, the inspection history of the facility, the novelty or complexity of the process, any application specific concerns, and we make an assessment as to whether we could cover the residual risk, for example, using a document request via the Section 704A4 of the Food, Drug, and Cosmetic Act, or if we could cover it using a remote interactive evaluation. So that is part of our decision process as to how to achieve our objectives. I would add to that we have access to information the facility might, so we don't really consider requests from companies to do remote evaluations, but that is a tool that is available to us that we do consider as part of our strategy for how we manage our inspections. Thanks.
Thank you for responding to that question. Alonzo Cruz, did you want to shed any light on that?
Yeah, I was just getting ready to say complete response. I don't have anything else to add.
Thank you so much. Moving on to our next question for Christopher Downey. And Dr. Downey, here's the next questions. Can you elaborate on the expectations that the product be in production during a PLI?
Yes, thank you for the question. That's a tricky one. So in general, it means that the product that's the subject of the BLA is being manufactured in the area and on the equipment that is described in the BLA. In general, we don't consider, looking at a different process area that's making the same product that's not yet ready for the BLA, for example, or not yet submitted, or surrogate products. So again, typically the expectation would be it's manufacturing the subject product of the BLA in the subject suite or line that's described in the BLA in the facility that's listed in the BLA, and that would be during the PLI period, and that would be occurring during the review of the application. Thank you.
Thank you for responding to that. We do have one more question here on PLIs, and it's the following question for Dr. Downey. Are PLIs pre-announced or might they be unannounced?
Thank you again for the question. The PLIs are pre-announced typically. I think the most recent PDUFA Act had a commitment to try to see if we can announce 60 days in advance, which I've always found kind of an interesting thing to ask, because it's known that PLIs are part of the process. So in a sense, you could say I'm announcing it right now. Like if you submit a BLA, you're likely to be subjected to a pre-license inspection. So it's the timing is generally worked out with the applicant initially, and then we communicate directly with the firm. But they're never a surprise. The timing is based on, because we observe the product in production, it's based on when they're manufacturing. So the company knows when they're manufacturing, when we've asked when we might be able to come, and then we communicate with the facility to work out logistics. So there's quite a bit of knowledge in advance, and they're not surprises. The facilities should be prepared. Thank you.
Thank you for responding to that group of questions. Moving back to our first panelist, Alonzo Cruz, we do have a few more questions that just came in, and here's the first question. Are there any plans for applying RRAs to CGMP inspections within the U.S.?
Yes, thank you for the question. I know it seems like we are using remote regulatory assessments exclusively internationally, but we have used them domestically. One of the earlier questions I had was which was more efficient and effective or better use. I forget how it was exactly phrased. And the physical inspection gives us all of the factors we're looking for into trying to make that CGMP assessment of the operating manufacturing facilities. Since domestically we can generally get to those places a lot easier, the percentage of time in which we would use an RRA would be minimum when compared to international travel, just due to other challenges associated with foreign travel, and if there are other advisories or other things preventing travel, or health warnings preventing travel to a particular part of the country. Thanks for that question.
Thank you for responding. And speaking of safety and traveling, we do have another question regarding that for Mr. Cruz. And can you speak to the agency's thought concerning safety of inspections in countries such as China?
Okay. Again, thank you for the question. Safety of our staff first and foremost is paramount. We took a lot of steps early on in the public health emergency to further ensure safety, and certainly continue to do so. For this audience, one of the many things that we look to is to the State Department and State Department guidance and warnings of particular challenges in traveling to particular parts of the country. We have resumed routine inspections in the country of China. As you may know, it was very difficult to travel in and around the country due to restrictions based on COVID. As of May of this year, we have resumed a more normal return to inspections in countries. That's including a staff that's located in the Foreign Office in Beijing, as well as staff traveling from the U.S. So we are resuming, slowly ramping up our inspectional activities within country. Let me also mention, because it's really top of mind on the news with the ongoing conflict in the Middle East. Immediately upon hearing of what was going on, there were a number of sites that we had to postpone travel to those particular facilities at this time because of the risk of travel in and around Israel. But outside of Israel and China, we're making these assessments on almost every foreign trip that we are going to, and making sure that we can provide the investigators can be safe while they're within country. Again, thank you for that question.
Thank you for responding to that question. And we do have just 1 minute, an opportunity for one more question for you, Mr. Cruz. And here's the question. Since you are in a MRA process with the EU, why are you conducting foreign inspections in Europe?
Okay, again, another great question. The MRA is intended, obviously, one of the main factors is reduced duplication of efforts within our respective inspectorates. It does not, there are inspections that, and it pertains to surveillance GMP inspections. There are going to be times in which we are responding to some signals we may have heard about, responding to complaints of injury or illness, maybe responding to a for-cause assignment in country. There are certain types that may fall outside of the scope of that. Now, if it is a inspectional activity that the EU member country can readily do for us, we may extend that reach for them to do those inspections. But the MRA wasn't solely intended just to prevent inspections within our respective countries. And the EU member countries have and continue to still at times conduct inspections within the US. Again, but thank you for asking that question.
And that's all the time we have for questions in our Q&A panel. A huge thank you to our presenters for answering numerous questions that came in. This takes us right into our break until 2 p.m. Eastern. Please enjoy your break. Welcome back from the break. We're moving right into session 2 on quality policy to provide an overview of policy document options, development, and oversight by Layla Weiser, who's the Director for Editorial and Project Management Staff in the Office of Policy for Pharmaceutical Quality in OPQ. Next, we'll learn about international harmonization, ensuring availability of quality medicine from Dr. Teresa Mullen, who is the Associate Director for Strategic Initiatives in CEDAR. And our final presentation before our next break is on ICHQ 12 implementation. What does industry need to know? By Commander Mahesh Ramanathan, Director in the Office of Policy for Pharmaceutical Quality in OPQ. Please join me to welcome our first presenter, Layla Weiser.
Good afternoon, everyone. I hope you had a great break. My name is Layla Weiser. I am the Director of the Editorial and Project Management Staff in the Office of Policy for Pharmaceutical Quality within the Office of Pharmaceutical Quality in CEDAR. Today I'm going to be providing an overview of policy document options, development, and oversight. Just want to take a step back for a moment. Something we really believe strongly in is that everyone deserves confidence in their next dose of medicine. So pharmaceutical quality assures the availability, safety, and efficacy of every dose. Today's presentation, I'll go ahead and provide an introduction of OPPQ, provide examples of policy documents that OPPQ manages, go through the common themes of the policy development process, and then really kind of focus on that. What does it mean to me? How does all of this fit together? Where does it fit within the regulatory framework? And then finally, we'll provide information on how to engage with OPPQ if you have any questions, comments, or concerns. So why OPPQ? We looked around and found policy offices existed throughout the other CEDAR centers, and they had different approaches to policy setting in those offices. So OPPQ was created to have a dedicated office with dedicated staff to focus exclusively on pharmaceutical quality. As you've probably imagined, pharmaceutical quality is a topic that touches all products regulated by CEDAR and CBER, and it touches all UFA programs. So it's a really large net and a very important one. This is a long list of the example of work products that we work on. These are anywhere from regulations all the way to individual policy to questions and issues. I won't read through them all, but it gives you a sense of the different groups that we tap into to be able to provide assistance in this space. So let's start off with guidance for industry and talk a bit more about that. I have provided the link here if you'd like to go to our public-facing page where you can search through guidances by topic. So what is a guidance? A guidance assists industry in carrying out its obligations under laws and regulations on subjects such as the content and submission of applications and the design, production, manufacturing, and testing of regulated products. You may not realize, but there actually are two different levels of guidances that we develop here within the FDA. Level 1 guidances provide FDA's initial interpretation of new or revised significant regulatory requirements. Then there's something called a Level 2 guidance, which usually addresses existing practices or make minor changes in FDA's interpretation or policy. When we're developing guidances, there are also two stages to development. The first one being development of a draft guidance, which describes new or significantly changed policy. They're published for public comment through a public docket, and they're not for implementation. Final guidances then is our next stage, and that's where we incorporate those public comments provided after a draft or a revised draft has been published. And because we're finalizing it and taking public comments into consideration and incorporating them, it's considered FDA's current thinking and therefore is appropriate for implementation. We also have non-binding versus binding language. FDA's guidance documents are generally non-binding. So when you open up one of our guidances, you'll often then see the text there in quotes, which talks about representing our current thinking on a particular topic, and that you also have an opportunity to provide alternate approaches if it satisfies the requirements, and to contact FDA to do so. But in rare cases where Congress has given FDA the authority to issue a binding guidance, we can use partially or fully binding guidances. And that means then that the guidance would include binding language. And just to give you an example of what we do when we have that binding language, the guidance on comparability protocols for post-approval changes to the chemistry, manufacturing, and controls information in an NDA and/or BLA has both binding and non-binding language. I've provided a link. Go ahead and take a look when you have time, and you'll see how we can go back and forth between those, but also what different language we use to set off the different types of requirements that we have. So another type of policy document that we work on routinely is a manual of policies and procedures, which we call MAPs. And I provided the link for that right here on this cover page. If you want to search for our MAPs, they are available externally. So what is a MAP? Policies and procedures primarily intended to direct CEDAR staff in the conduct of their work, so directives, and they're going to be published in a MAP then, and it serves to disseminate the policies and procedures. Most MAPs are posted for public view for the purpose of transparency, but unlike guidances to industry, MAPs are really not intended to provide guidance or to gain public comment from the regulated industry. We do have MAP categories and different types of review. For categories, we have a standard MAP, an interim MAP, and then internal MAPs. And the type of category really then it's intended then to determine whether it's for internal or external audiences, and that really is driven by content of the document. And we have review cycles as well. The MAPs are periodically reviewed to ensure continued relevance and effectiveness. And then a third type of document that we work on is called a compliance program. Again, I've provided the URL if you want to take a look at the programs. Although this is intended for our internal audience, it's something that we provide transparency to and post externally. So what is a compliance program? It provides instructions to FDA personnel for conducting activities to evaluate industry compliance with the Federal Food, Drug, and Cosmetic Act and other laws administered by FDA. And so while we have different types of policy documents, the life cycle that we use to develop these policy documents have many similarities. So this is an overarching structure of how we would go about developing policy within our office. We first need to focus on prioritization. As you might imagine, there are competing priorities throughout. So we really need to first consider then statutory priorities, but then also UFA, public health emergency, agency, and center priorities. So it's not something office specific or something that we're personally interested in. This is a holistic review of what needs to be taken care of to ensure public health. We then form a working group, and that's where the policy writing and development happens. And once that document is developed, it goes through clearance and then ultimately publication. Then we need to also consider, once it's been posted though, how about implementation? We need to consider whether staff need training or what sort of communication we need to share with our colleagues in industry and other external stakeholders, but also internal business partners. But our job isn't done because we want to continue to revisit that policy that we put out. So we have a policy document review process which ensures periodic review of our documents to ensure continued relevance and effectiveness. So I mentioned at the earlier stages here about understanding the regulatory framework. We work on all these different documents, but how do they all fit together? So let's start with regulations. Our regulations lay out policy requirements at a really high level. So, for example, not every testing method would be discussed in a regulation. But when you look at a guidance to industry, it provides FDA's interpretation of the statute and regulations to provide recommendations on specific topics. So the example I provide here is extractables and leachables. And then we look at MAPs and compliance programs, which is our opportunity to internally operationalize what we have stated in guidances and regulations. So the really important thing here is reading those together is how you can get a better understanding of FDA requirements and recommendations. I'm going to provide some examples for you within the regulatory framework, how one document type might be a little different than another. So the topic we're going to look at for this first example is about allowable excess volume and labeled vial size in injectable drug and biological products. We wrote a guidance for industry to provide recommendations for industry to comply with the CFR for injectable liquid, solid, or reconstitution drugs, while mitigating inappropriate excess volume. So the guidance we use then is used to understand the relevant regulatory framework, and that is applicable regulations and FDA's recommendations on how to address this issue in your application. And you'll see in the example here that the guidance clarified our regulatory requirements and recommendations, and then described when justification is needed for proposed excess volume. And then it discussed the importance of appropriate fill volumes for injectable drug products and provided recommendations for fill sizes. And I provided a link here so you can go ahead and take a look at that document on your own time. But let me move on to the next slide. And on this slide then, again, same topic, but now we have a map written about this topic. It's called MAP 5019.1 Revision 1, which describes how CDER OPQ will consistently assess fill volume for injectable drugs. So here you're intended to read the map as an internal reviewer, but also those of you are able to review it externally, because I mentioned it's posted externally. But you're going to read that map to understand what OPQ assessors will be looking for in your application. And you can use this map then to double check your understanding of the guidance. So in the map, we describe policies and procedures for product quality assessors to ensure consistent assessment of excess volumes. We also address liquid, solid, and drug products that require reconstitution and constitution. And then finally, we describe the policies and procedures to be used by OPQ product quality assessors to ensure the drug product is sufficient to allow for withdrawal and administration of the net container content of the drug products. You can go, we take that deeper dive, and we really provide that understanding to our assessors of what to look for. And again, for that, then I've provided a link to that map if you want to take a look at that and see the distinction between the two document types we provided as examples. Let's go over a second example with a different topic. This topic is process validation for drug manufacturing. We're going over the different types of statute and regulations relevant to process validation. We talk about 501 and then 21 CFR and 211. If you take a look at those, I won't read through them because they can be a little clunky to get through. But you can see these relevant provisions of FDA's statutory and regulatory authority to understand the relevant requirements. But when you move on to guidance, again, now we're on example 2, the same topic of process validation for drug manufacturing. We developed a guidance on process validation, general principles and practices in January of 2011. That specifically outlined general principles and approaches that FDA considers to be appropriate elements of process validation, and then incorporated principles and approaches that all manufacturers can use to validate manufacturing processes, And then it allowed you then to align process validation activities with the product lifecycle concept and relevant ICH guidances. To point to this being that you would use the guidance to understand more fully how to design and implement process validation activities. Like the other slides, I've provided a link here if you want to take a look at that guidance to get a sense of the tone and the approach used to provide that clarification. And in this example, we even have an example of a compliance program for this same topic. So the compliance program here is about post-approval inspections. Remember, this is an internal document for our FDA personnel. It describes how FDA will assess, among other objectives, CGMP compliance regarding process validation lifecycle activities for approved drugs. And then it provides risk-based strategies for the scope of inspectional coverage and clarifies roles to establish efficient communication. So boiling that down, you read this compliance program, even externally, although it's intended for internal staff, again, it is provided externally for transparency, but you would read this to understand what process validation information FDA investigators may evaluate when inspecting your manufacturing facility. So a very, very useful tool. And again, collectively then provides a really great overview of FDA's expectations and hopefully can clarify then what your understanding is of our needs. So I want to provide some final thoughts. So the big thing is here, remember, when you're looking at a draft or finalized guidance, also look for a related map or compliance program to provide further insight into the guidance. You saw these examples that we just went through and what a build it is, and that there's different layers. It's like a tapestry to be able to better understand the topic and our interpretation of a topic and how we're advising staff even to consider that topic. Maps and compliance programs, that's how we operationalize our interpretation of a rule or regulation. So they're really going to help understand FDA's practices and how FDA ensures consistency. These policies at the end of the day, going back to that first slide about what is important to us, they're intended to be understood and used to ensure our goal of safe, effective, quality products. So how to engage with us? We have a dedicated email address that you can send any questions or comments to. It's cedar/opq/inquiries@fda.hhs.gov. We take comments and questions from all of you very seriously and make every effort to be timely and thorough in our responses. I really just also want to take a moment to thank you all. This has just been a great opportunity to strengthen our partnership with you. I hope that this particular presentation has afforded you the opportunity to see what these documents' purposes are and how collectively we use them to really frame that regulatory framework. Thanks so much for your time, and I appreciate it. And back to you.
Hello, I'm Teresa Mullen, and I'm going to talk today about enabling pharmaceutical quality management through international harmonization of standards. Well, effective pharmaceutical quality management is necessary to ensure against quality failures, supply disruptions, and drug shortages. And it typically requires post-approval changes to be made to regulatory filings because facilities age, routine operations may require updates to maintain GMP compliance, regulatory requirements change, and technology evolves, suppliers may change, and companies may generate new knowledge about products and processes. And regulators around the world want to encourage effective pharmaceutical quality management to enable companies to make those changes and keep making those innovating and applying what they learn in commercial operations to continually improve. But post-approval changes can require separate prior approval from each regulatory authority where the product is marketed. And that can create complexity. Now, for one regulator like FDA, that picture on the top here may be what we see, which is we get a post-approval change submitted, and we're doing our review based on our timeline, and we provide a response back. But for a company, it might look like that picture on the bottom, where they're submitting that post-approval change to multiple different regulators who may even have different requirements and different timelines, come to different conclusions, and the company needs to wait to hear from all the parties where they've submitted in order to proceed. And this can create some delay, uncertainty, and in some cases it may discourage a company from doing what we want them to do with managing quality. So regulatory reliance ultimately could help with this, reducing this kind of complexity. But we can even begin to reduce this kind of complexity very critically by harmonizing the regulatory requirements in different regions, so the same things are expected by regulators in different regions. And this then would provide a comparable basis for our regulatory assessments, which then could enable, if other conditions are appropriate, to actually rely and use another's work product, rely on and make use of another regulator's work product or their assessment or their inspection report to support and inform our assessment of post-approval changes. Of course, companies would need to be receiving the same quality dossier for that to happen. But foundational here, and that's why I have it red, is that harmonization of regulatory requirements is really key to enabling these things to happen. And ICH is where we do this work. So the International Council for Harmonization of Technical Requirements Pharmaceuticals for Human Use, this is why we just call it ICH, is a unique harmonization organization that involves both regulators and industry. It was launched initially in 1990 by the US, EU, and Japan. And it had a well-defined objectives which hold true to today, which is to improve the efficiency of new drug development and the registration process, really reducing duplication, aligning standards, and getting rid of unnecessary differences, promoting public health by, for example, reducing the duplication of clinical trials and unnecessary animal testing that isn't really needed to generate that evidence that we need of safety and effectiveness. And this is accomplished through the development of these harmonized technical standards and guidelines, and then the requirement that regulators in different regions will implement those guidelines. Although it started out as having 6 members, 3 industry, 3 regulators in those founding regions, today we have 21 members and 36 observers at ICH. It's really a truly global operation. And with the regulators, if you think about how powerful this is, that regulators committing in all of these different regions to implement these guidelines. So ICH has produced quite a few. This is as of last year. There are a few more now, but as of November 2022, 72 guidelines in different topic areas, 16 guidelines in the safety, that's animal safety studies primarily, 25 guidelines in the quality space, 22 for efficacy, and 9 in the multidisciplinary area. That includes electronic standards for the transfer of regulatory information, notably the electronic common technical document, modules 2 and 3 being where we have the quality information, and MEDRA, which is the standardized terminology for adverse event reports. So how are these guidelines developed? Well, there's a 5-step process that's pretty standard for all the guidelines. After a new topic is approved by the ICH Assembly, an informal work group is established, and they develop a concept paper which outlines the scope of what will be done in this guideline and the time frame in which they anticipate doing it. And that is after being approved by the management committee, and that document, that concept paper, then will provide the basis for the group to be established and the work that they're going to undertake. The first step in this process is consensus building among the industry and regulatory experts to produce a technical document. When that's produced, it's forwarded to the assembly for approval, first by all parties, and then a second piece of that is just by the regulators to review and adopt it. And why does it switch to just regulators? Because at the end of the day, this recognizes that the regulators are the ones that are going to be implementing these guidelines, and they need to own it in order for that to happen. And so then it goes into a step 3, which is really the public consultation or public comment period in our FDA parlance. And after the comment period closes in all the regions where that's being undertaken, those comments are gathered up. the group reconvenes to go through all the comments and address them in the revised guideline as needed, and that results in a Step 4 guideline, which is the equivalent of FDA's final guidance. Following that is the implementation step, which is very critical to the effectiveness of ICH at all, because if we don't implement consistently in different regions, we won't get that benefit of harmonization. And so training is really critical following that implementation stage to ensure that there's correct and consistent implementation of the guideline around the world. So in the quality area, we have a number of really critical guidelines. And the shorthand names here may sound very familiar to many people. And so overall, these guidelines focus on pivotal milestones like the conduct of stability studies, defining the thresholds for impurities, flexible approach to managing quality, product lifecycle management, continuous manufacturing, and so on. And I'd particularly draw your attention to the Q9, quality risk management, Q10, pharmaceutical quality system, and lifecycle management for the quality management topic. Now, there's an annex in Q10 that really lays out the progression or increasing levels of quality management maturity that a company might basically demonstrate, and how that translates into a more risk-based and flexible approach to regulatory oversight, starting with just basic GMP compliance, which is kind of the status quo and minimal. A company might go on to having an effective pharmaceutical quality system, PQS. They may further demonstrate product and process understanding, and so on. And there's a progression there. Well, this guideline was put together in the 2008-9 timeframe, and there was not as much uptake of it as was hoped. And so Q12 was in part a response and a further sort of addressing of this need and opportunity. Now, Q12's full name is Technical and Regulatory Considerations for Pharmaceutical Product Lifecycle Management. We refer to it as Product Lifecycle Management, provides this framework to facilitate the management of post-approval changes in a more predictable and efficient manner. And really, how can increased product and process knowledge help reduce the number of regulatory submissions? Providing very explicit tools and enablers to kind of operationalize, you might say, what was in the annex of Q10. And notably, a company coming up with established conditions, the post-approval change management protocol, which lays out the strategy, product lifecycle management, and so on. So, in summary, effective pharmaceutical quality management requires a lifecycle approach, and it includes post-approval changes to regulatory filings. But many post-approval changes require prior approval by each authority where the product's marketed. This can generate regulatory complexity and operational challenges. It's burdensome for regulators and industry. And ICH works to harmonize the technical requirements for quality management and those regulatory submission formats. So these involve Q and M guideline work. ICH harmonization work is undertaken by regulator and industry experts, and it can take several years of pretty intensive work to produce a consensus guideline. But the resulting guidelines enable regulatory consistency and efficiency, and they reduce the uncertainty and risk for companies trying to manage quality. An effective management of quality is really necessary to ensure the availability of medicines that are critical for patients, and this is true globally. And with that, I thank you.
Hello, my name is Mahesh Ramanathan, and it's a real pleasure to be talking to you today on FDA's implementation experience with ICH Q12. And hopefully today, I'll be able to walk you through the foundations of the guideline and the guidance document for FDA, how to utilize some of the tools most efficiently, and also share with you what we've been seeing when it comes to the use of ICH Q12 and submissions and their benefit to the application overall. All right. So you've seen this already, but I'm going to put up here just for fun anyway. Let's go ahead into the actual content of the presentation here today. As a reminder, hopefully you all have read ICH Q12. It has been out for 4 years now, and our implementation guidance has been out for 2 years. Hopefully you are all familiar with that. And if you're not, I highly encourage you to read those 2 documents as they really paint a broad picture of how to use ICH-Q12, its goals, what it intends to achieve, and best ways to approach the tools. But for those that have not read it, ICH-Q12, as we call it, Technical and Regulatory Considerations for Pharmaceutical Product Lifecycle Management, really utilizes all the best tools in Q8 through Q11 to provide a framework to facilitate the management of post-approval CMC changes in a more predictable and efficient manner. And really, the take-home here is as applicants better utilize the tools that we have in ICH Q8 through Q11, it provides the opportunity to present scientific justifications to reduce the volume of elements that would be CMC changes that require a filing to the agency, and offer the opportunity to reduce the reporting categories that go along with it. So overall, less things that need to be reported to the agency at a lower level if the tools are used correctly. And really that's summarized in that 3rd bullet there, that when the Q8 through Q11 tools in the Q12 manner is used in a stronger fashion, opportunity for fewer established conditions. When it's weaker or less robust, it's likely going to be more established conditions for the applicant. Important note here is that the use of ICH-Q12 is voluntary. Implementation is also flexible in the sense that the proposals for specified established conditions on a product lifecycle management document can happen at any point in the life cycle. It can happen with the original application, or it can come in as a post-approval supplement to identify and define the established conditions for a particular application product. It's also flexible in the sense that it can be used for as little or as much of the CMC sections of the application as the applicant desires. For example, it can be defining these ECs for one specific unit operation, if that is where the applicant chooses to invest their time and resources in deploying ICH-Q12. It can be for everything in Module 3, from your drug substance and drug product, manufacturing processes, container closure systems, analytical methods, manufacturing facilities, etc. The flexibility spans that spectrum. Okay, a little bit on where we are with ICH-Q12 from an FDA perspective, just to paint the overall picture before we go into more detailed discussion of the tools themselves. So we adopted ICH-Q12 in 2021 as a Step 5 document, and that replaced our 2015 draft guidance on established conditions. That guidance was no longer necessary as ICH-Q12 became reality, and we can now point to that as official FDA policy. In parallel to the adoption of ICH-Q12, the parent guideline there, we also published FDA's implementation considerations draft guidance document in 2021. And the goal there was to identify and clarify how applicants working in the FDA system can translate the terminology, translate the approaches to use here in the United States, as well as accounting for some specific frameworks to the FDA system that still offer the opportunity for using ICH-Q12 tools. So for example, using them in the frameworks of referencing a drug master file, using a drug device combination product, and how to approach established conditions for those types of drugs, etc. And finally, what we are working on is a manual of policies and procedures, which is going to really focus on specific procedures for the assessors on how different disciplines encountering ICH-Q12 tools, specifically proposals for established conditions and reporting categories for those established conditions, are going to work through the assessment, both from the application side of things and the assessment of facilities in the pharmaceutical quality system side of things, and how those come together to make initial decisions in support of applications, as well as to maintain or how to respond to maintenance after approval of those particular applications. Okay, so the scope of ICH-Q12 itself, this is a summary of the guideline, so I won't read it verbatim, but the real take-home message here is that when it comes to ICH-Q12 and the FDA, we've had no limitations on the scope. We are fully implemented across anything that Q12 can be deployed to per the guidance. We are open doors for those approaches for applicants. All right, and similarly, we have a number of tools in ICH-Q12. And the point of this slide is not to go through every single one in detail, but to really highlight that through these numerous tools that we have in ICH Q12, the applicant really should be looking at these as a suite. How to utilize all of these in conjunction or to the best of their ability to really open the door for maximum opportunities when it comes to justifying fewer established conditions and lower reporting categories. Again, the real take home here is that within FDA, we have fully implemented ICH Q12, so all of these tools are open for business. And again, coming back down to the applicant to decide how to best approach it and pursue maximum benefit. I'm not going to spend time today going through every single one of these, but I did want to bring particular attention to established conditions, PACMPs, or as we call them in the United States, comparability protocols, how it all works together in the product lifecycle management document, and then the pharmaceutical quality system being that underpinning and that foundation of efficient implementation of Q12. So what are established conditions? If you haven't read the guidance documents, either the parent ICH Q12 or our implementation considerations guidance document, this is a very important time to pay attention and listen to what ECs are. So ECs are legally binding information within the application considered necessary to assure product quality. As a result, in the context of ICH Q12, as a result, any change to an established condition would require reporting to the regulator. Now, the big sort of take-home message here is to recognize that an application today is a mixture of established conditions and supportive information. And some of those established conditions examples that we all see in front of us, could be the actual drug substance, the drug product formulation, the manufacturing process and its controls, the specifications, the facilities, etc. Not uncommon, and hopefully is not news to anyone hearing this presentation, that when these elements are changed, there's an expectation for reporting to the application, whether it's a prior approval supplement or changes being affected, etc. Maintaining the file to account for what's currently going on in any one of these particular areas that are established conditions. But not everything in the application is an established condition. For example, the data supporting method validation, or the data supporting stability, or the data supporting process validation or process development approaches. Those things, when they are maintained, don't themselves necessitate a filing to the application to gain approval from the regulator for any sort of change. We would consider that more supportive information. So the concept of established conditions, which exists today in the regulatory frameworks, is really to highlight here that the application is a mixture of both. But when you change the established conditions element, that file will have to come to the regulator for either a notification or approval. A real important message, though, when it comes to Q12 implementation is that all changes, no matter whether they're reported to the application or not, require a change management approaches under the pharmaceutical quality system. Okay, so when it comes to Q12 implementation, I kind of mentioned this early on, but I'll come back to it again in the sense that the extent, i.e. the number and how narrowly defined of the established conditions, will vary depending on multiple factors. So how much product and process knowledge does the applicant have? What way do they gather it? How can they use that information to justify their proposals for established conditions? And their grasp and kind of mastery of demonstrating or showing the potential risk to product quality if that element were changed, given their product and process understanding, etc. Now, after identifying the established conditions, applicants can propose various ways of managing those established conditions, i.e. how they would report them to the application. A lot of flexibility here as well. Applicants can propose and choose to follow existing regulations and guidance. For example, here is a list of established conditions, but when changing them, I'm just going to follow, rather the applicant will just follow 21 CFR 31470 if you're a small molecule product, or 21 CFR 60112 for a large molecule product, and any implementing guidances on the types of reporting categories for specific changes, for example, the SUPAC guidances. Alternatively, an applicant has the opportunity to propose different reporting categories based on their knowledge of the product and process and their knowledge of risk when managing a particular change. For example, an applicant can say and propose in their submission that even though SUPAC would have this particular change reported as a prior approval supplement, the development knowledge that's been gained, the platform knowledge that's available, as well as the risk management assessment of this particular established condition and its link to quality attributes holds that we can change it, or rather it can be changed with a lower level of risk than what is normally expected or normally considered to be the level of risk. Hence, let's report this as a change is being affected or even an annual report instead of a prior approval supplement. Those are opportunities that are available to the applicant depending on their desire to pursue those proposals. And I'm happy to report that we have seen quite a few of them and have approved quite a few of them, dependent on justification provided by the applicant. Okay. Now, this is a figure that we have from ICH Q12 itself, and I've always found it to be a fairly good picture how to work through the thinking of what I just talked about in the previous slide as far as identifying the established conditions and then the reporting category. And the way that I think about it is above the red line there, those questions and outcomes are geared towards is the thing an established condition. Below the red line is, well, for that thing, if it were changed, what would be the reporting category? And that top element there, this is a figure specific for manufacturing process parameters. It can be applied in its thinking across different elements of CMC. For example, the performance characteristics or primary performance characteristics for a device constituent part. In our implementation guidance, we have an adaptation of this figure to combination products. It can be applied to analytical methods and really focusing in on the characteristics or elements of that method that assure performance of that particular method. But here with process parameters, just to flesh it out even more, when we talk about parameters, looking at whether or not that parameter needs to be controlled to ensure product quality, and that can be thought of in a few ways. One, is it a critical process parameter, or is it a process parameter where the impact on quality cannot be reasonably excluded? There's not enough information yet, or there is some signal of a potential relationship, however more work needs to be done. And depending on the answer, yes or no, it could be determined an established condition or not an established condition. If the answer to those questions are no for this particular process parameter, it could be reasonably justified as not an established condition, in which case it's not reported to the application. Now, that's not to say it's not managed, and that all changes must be managed by the pharmaceutical quality system. It just provides the opportunity where that particular change doesn't require an annual report or a CBE or a PAS and the waiting for approval from the regulator. On the other hand, if you are an established condition, it will be reported to the application if changed. And the second half of this diagram walks through how to apply Q9 type principles in considering the output of the criticality assessment and the control strategy to identifying the potential risk to product quality if that particular parameter was changed. Higher risks would trend towards prior approval. Moderate or lower risks would trend towards notification type changes, which in our FDA system would be your CBEs and your annual reports. Okay, now I previously talked about how the difference in development approach or product and process knowledge and understanding available or platform knowledge available could lead to differences in the established conditions. And what we have here is a screenshot from the ICH Q12 implementation working group training materials, which are available on ich.org. If you search down and scroll to ICH Q12, there will be a link to our training modules. And this is a screen grab from that particular one of those presentations, excuse me. But what it shows here is that depending on the type of approach, whether knowledge is gained in a very superficial fashion under a minimal parameter-based approach, contrasted all the way to the right side of the screen, where there's a knowledge-rich environment of the interaction of parameters to quality attributes, such that you, and potentially even using technologies where there's online controls and ways of feeding back information from the process into the control of that particular unit operation. Consider that a performance-based approach where there's maximal knowledge and control of the process itself. And what you can see is that now these are not meant to be three distinct approaches as far as the only ways of doing things, but they are meant to illustrate a spectrum. of minimal knowledge to maximal knowledge. And along the spectrum, if you look left to right, you will see that the things that are established conditions change, as well as the reporting categories associated with them change, to where on the left side you have more established conditions requiring filings to the application have changed. For example, look at your equipment type, your V-blender. It is an established condition, and if it were changed to a different type of equipment, it would be at least a CBE 30. Moving across the spectrum there, a situation where there is enhanced knowledge under a parameter-based approach, that risk might come down to a lower notification, notification low, potentially a CBE 0 or an annual report, to the other end of the extreme under performance-based, where perhaps the applicant is able to justify that the type of equipment doesn't even matter anymore, and it can be controlled as far as the quality of material can be controlled independent of the type of equipment, to where the applicant has full flexibility to change that equipment without reporting it to the agency. So let's shift gears a little bit here from established conditions now into another tool that's available in ICHQ 12, what we call the Post-Approval Change Management Protocol, or PACMP. In the US system, PACMP is the same as a comparability protocol. Please look at our current guidance on comparability protocols, and you'll find extreme congruence with the content that you see in ICHQ 12. But for the purposes of the presentation, I might use PACMP and CP interchangeably. If I do, I'm talking about the same thing. So a PACMP, what it does is provide predictability and transparency in terms of the requirements and studies needed to implement a change. And if those requirements are met and studies are conducted as expected in that change management protocol, providing the opportunity for a lower reporting category when implementing the change. The beauty of a PACMP is that it can be utilized for one or more changes to a single product, or maybe used across products when that change is common, and the characteristics of that change is common across products. For example, potentially a simple liquid formulation injectable product utilizing a container closure system, glass vial and stopper. Think about the opportunity to change the glass vial and stopper for multiple products at the same time using the same protocol. PACMP, much like established conditions, has extreme flexibility in when it can be proposed to the regulator, and that can be submitted with the original application or subsequently as a standalone supplement. Okay, so what could that look like with a PACMP? So with a change management protocol, step one will usually involve the submission of the written protocol in the form of a prior approval supplement to the regulator, and that will go over the tests, the studies, the acceptance criteria, etc., where the regulator can then look at the plan, look at the proposed outcomes of the plan, and provide feedback or concurrence in form of approval to that particular supplement back to the applicant. If approved, we now have that transparency built in on how to approach a particular change between the applicant and the regulator. Step two is then the applicant actually carrying out the tests and studies in the protocol, and if everything goes as expected, the results, the data generated meets the acceptance criteria, any other conditions specified in the approval of step one, that information will then be submitted to the regulator pursuant to the reporting category agreed upon, usually a lower reporting category than what would normally be expected, and can go forward with implementing that change. This can be done potentially at a notification type level, for example, a CBE or an annual report, giving more flexibility for streamlined and quicker change implementation. However, depending on the category, there may be situations where certain changes might need to come in at a prior approval supplement. For example, if the protocol acceptance criteria were not met, or if significant changes had to be made to the protocol, or if the risk of the change was agreed upon to be just too high to necessitate a notification or support a notification. Okay. So you might be asking yourself, what's the difference then between established conditions and a change management protocol? In reality, they are fairly well aligned and pretty close together, and they all share some common characteristics, which you can see on the first three rows there. A major difference is that with a change management protocol, you are getting the alignment up front on the approach, the studies, and acceptance criteria, and defining those up front to the regulator in the form of a submission of the protocol to the application. With the established conditions, it's a much wider open sort of approach in the sense that the applicant is justifying of what would need to be reported and at what level, based on the available either development knowledge or platform product and process knowledge at the time of that submission, and giving flexibility through that sort of mechanism. All in all, this all comes together in the product lifecycle management document, or the PLCM, as it's called in ICH Q12. And the PLCM is a central repository of the established conditions, the reporting categories, details of the established conditions. For example, if it's a process parameter, the range of that process parameter, or the controls around it, specificity in the reporting categories. So not only one, will it follow US regulation and guidance, or is it something different that's proposed? For example, a lower reporting category, but also clarity on potential situations where the reporting category could be different. For example, if going beyond the lower end of a range is a lower risk, that might be a lower reporting category than going beyond the higher end of the range, that might be a higher risk and a higher reporting category. So directional based reporting categories having an opportunity in the PLCM as well. Now so all the ECs in the reporting category should be proposed and transparently captured on the PLCM. After approval, the applicant should be maintaining the PLCM with an updated list every time they make a change to an established condition. As well, from a pharmaceutical quality system management perspective, looking at opportunities to identify changes to ECs overall, those should be provided to the regulator too. So I mentioned early on about our draft guidance, ICH Q12 implementation considerations for FDA regulated products, and kind of what that does. And I'd already mentioned sort of how to use the ICH Q12 tools in our system and accounting for our frameworks, but the guidance does go into specific considerations that applicants should be aware of to be most effective in utilizing Q12 in the FDA system. So for example, clarity in the cover letter regarding proposed ECs, to be as explicit as possible that this submission contains proposals for established conditions and a PLCM in module 32R in the application, for example. Clear and specific identification of the ECs in the PLCM, the reporting category and justification. So, being as black and white as possible in the PLCM about what is the established condition, what is the reporting category, and not being vague about it, and clearly indicating the facilities in the PLCM, clearly indicating where those established conditions will be implemented. For example, if we're talking about established conditions for the drug product manufacturing process, to clearly concatenate any established conditions associated with the manufacturing process under the facility identifier for the drug product manufacturing operation. So we had briefly talked about kind of the PQS being the foundation of ICH Q12 implementation, in that while only some changes will be reported to an application, all changes are expected to be managed by the PQS. And kind of diving into further about why this is so foundational to Q12 implementation, in that the effectiveness of a PQS really provides confidence in the proposals for established conditions by allowing the regulator to see that there's a feasible and robust control strategy, that we have competent quality oversight for maintenance activities post-approval, that we are confident that all relevant data has been assessed and considered, anything that's been generated by a particular facility, and that the change management procedures are competent. We go on to talk about in ICH Q12 about how an effective change management process across the supply chain, across facilities that are linked up together in making a particular product is essential, because we really can't be thinking about the risk of changes within the silo of one particular element. It really needs to be broadened out and at least account for upstream or downstream impact. And I mentioned this on the previous slide about maintenance. Really, knowledge gained during the commercial phase is a key element of that post-approval confidence. That even after approval of PLCM and the appropriate, the established conditions, that as new knowledge is gained during manufacturing operations or changes in suppliers, routine stability, etc., that there is a periodic reassessment of criticality and risk, and searching for any needed changes to established conditions, which goes both ways in the sense that an applicant might learn something that gives them the opportunity to reduce the established conditions, to remove something that was previously thought of as a risk, but maybe is no longer with the additional knowledge gained. Alternatively, the opportunity to add established conditions for better life cycle control if new risks emerge in the context of routine operations, etc. This diagram here is from ICH Q12, and it really puts into a picture the relationship between the pharmaceutical quality system managing changes inside of a particular facility or at the applicant level managing changes, and on the red part of that diagram, the linkage up to established conditions and notification. So you see that the change management lifecycle is really agnostic to whether or not it needs to be filed to the application, and that all changes are being sort of managed under a consistent approach. And then in certain cases, the red dotted line over to the red box, in certain cases, some of those changes need to be reported to the application. What Q12 is doing is essentially making that red box smaller, or giving the opportunity for applicants to justify a smaller red box than what would be normally expected under regulation and guidance. But all of it kind of really depends on the effectiveness of the PQS, and knowing that this cycle, this approach, is going to be well implemented. Okay. So thinking about pharmaceutical quality systems, what would it mean for inspections? So if you are an investigator, or if you are if your facility being inspected, when it comes to Q12 implementation, there's no expectation for investigators to review established conditions. That will be done through the application with the assessors at headquarters. However, the inspection is an opportunity to gather information on the effectiveness of the pharmaceutical quality system. And for those that are paying attention, you will notice that we have updated our surveillance compliance programs and our pre-approval inspection compliance programs to add content about advanced pharmaceutical quality systems, about the change management effectiveness, et cetera, that in part supports ICH Q12 implementation. So as investigators looking at kind of these other areas of the quality system, particularly change management, it will help gain more evidence and more knowledge and more insight that can further drive flexibilities in Q12 proposals from applicants, or at least our assessment of them. But important to also know that if an inspection identifies a violative situation in the facility, it may impact the approval established conditions. It may also impact the viability of previously approved established conditions, to where changes might need to be reported at normal levels under regulation and guidance until such time as that facility is out of a state of violative compliance status. All right, so what have we done to get ourselves in a position to implement Q12? Long story short, we started 5 or 6 years ago on training FDA assessors and investigators on what ICH Q12 is, how to think about the change in thinking when it comes to letting science and risk drive some of the proposals for established conditions. That went all the way from theoretical examples to the output of the pilot assessments that we did in 2019, all the way through implementation of Q12 and the implementation considerations guidance. As I mentioned earlier, this also impacted the inspection compliance programs. This training and this exposure, was available or deployed to our assessors in the Office of Pharmaceutical Quality, our colleagues in the Office of Regulatory Affairs doing the inspections, as well as colleagues in the Office of New Drugs and Generic Drugs for awareness on what Q12 is and what it could mean to applications as they come in the door. In the day-to-day operations, now that we've opened the door and applications are coming in, we found the need to ensure continuity in the assessment in the early stages, especially as assessors are really starting to see submissions in real time. So we stood up 2 groups, the Established Conditions Coordinating Committee and the Q12 Assessment Implementation Team. The ECCC is really there to provide regulatory support to the assessment teams, helping them think through the policy, think through congruence with ICH Q12, and ensuring that any decisions or communications or language that we send back and forth to the applicant is supported by that policy. Whereas the Q12 Assessment Implementation Team, members there, represent our senior scientific folks across disciplines. We're there to help the teams really think about criticality assessments, risk assessments, and whether or not proposals for ECs are well justified. In addition to those two groups, a member from each one of those two groups joining the assessment team, we will also have a new discipline, a pharmaceutical quality system assessor, join the team to really look at those facilities where ECs are going to be implemented and whether or not there are any concerns with pharmaceutical quality system health, change management effectiveness, et cetera, that could influence decisions around established conditions and reporting categories. So here's a snapshot from the pilot implementation through September of 2023 of the types of applications that have come on board and sort of where we are with status. And as you can see, the concentration is really in the innovator space. And at this time is really in the supplement space too, where we're looking at proposals coming forward for established products, have been approved fairly recently, and prior approval supplements are now coming in to define a list of established conditions and reporting categories. A lot of activity in the biologic license application space, in the new drug application space, but not a lot of activity in the ANDA space. Where we are right now is a lot of supplements coming in. We're starting to see things start to tip over into more original applications coming in the door to actually build in this flexibility on the day of approval. Which is a good, I think, sign for things to come. But again, this is designed to be flexible, so it can happen at any point in time in the life cycle. For example, if a business model holds that it's not viable to do this at the original application stage, but it's better after there's an established product, that door will always be open for applicants to come in with PASs after approval and reduce their supplement burden and implementation in a global space. The types of ECs that we have seen, kind of the flavor of them. So we have seen everything from proposals to reduce the volume of volume of established conditions, where reporting categories are consistent with the regulation and guidance. We've also seen, actually the bulk of the proposals where the reporting category proposals are more flexible than regulation and guidance. And it's been really nice to see the supporting justification for both the established conditions and the reporting categories, which by and large have left us in situations where we can approve these applications. We've seen a mix of the types of ECs, whether it's a parameter-based approach or a performance-based approach where there's a lot of knowledge, maybe some feedback mechanisms, et cetera. But happy to say we've seen this across the spectrum for both manufacturing processes, analytical methods. We're starting to see proposals for device constituent parts of a drug-device combination product, and a very positive element as far as applicants proactively specifying by facility where the established conditions will be implemented. For example, we've had a few applications with dual facilities doing the same operation for a particular product, and the ECs within each facility are a little bit different by virtue of differences in equipment, differences in operations and controls, but nonetheless having that clear specificity on the PLCM allows for confidence in the approval as far as what we are approving and the ability to drive the PQS assessment. So our reflections, the next couple of slides are going to go over things that we have seen, also kind of allude to common information requests that we're sending back to applicants to get clarity on particular proposals. But reflections from our initial experience, really what helps Q12 implementation, or at least the submissions for Q12, are clarity in the cover letter, so that the agency can start off immediately in assessing these applications with the right teams. I mentioned on a previous slide that we have 3 additional members of the team joining when an application includes proposals for ECs and alternate reporting categories. In order to get them on board at the right time, seeing it up front in the cover letter is incredibly important. So whether that's proposing ECs or revisions to already approved ECs, or as simple as making changes in accordance with approved ECs, it's important to have that kind of clarity so we can tailor the teams appropriately and maximize the time in assessing these applications. Scientific justification is a huge element when it comes to the success of Q12 applications and their approvability. Really, looking at these 3 sort of bullets here, the justifications really need to focus on explaining the approach to criticality assessment. early on we got an inkling that some applicants were worried that what was in Q12 would be calling them to change their company approach to risk assessment or criticality assessment to conform to what was there in Q12. And that's not the ask. The ask is a clarity in explaining how an applicant went about criticality assessment and risk assessment so that the teams, when they're assessing these proposals for ECs and reporting categories, has the contextual understanding about why the applicant is proposing, a certain level of criticality or a certain reporting category level to then frame out whether the justification makes sense. So the ask is not to change, the ask is to explain so that the assessors know what they're looking at. Again, that's the second bullet there, right? That the justifications and risk assessments should clearly describe the scientific rationale for why something is considered an EC, or in some cases, why something that would normally be considered an established condition is not in this case. Same thing for the reporting categories. And one thing to highlight here, especially if an applicant is considering filing a PAS to document established conditions or gain approval for ECs for an already approved application, is to really ensure that the justifications for those ECs and reporting categories are current and are actually justifying the ECs and the reporting categories and not just the parameters that were approved for the process. In more than one situation, we saw justifications that were pointing back to data or studies conducted for the original application approved, 5 or 10 years ago, to where even the thinking around established conditions didn't exist. The justifications didn't really make sense. So really think about when you are proposing ECs and reporting categories that are using the principles in Q12 and the implementation guidance to frame things in the right way to support justifications for why this particular element should be an established condition, why the reporting category should be at this level, etc. Okay, some more reflections. The PLCM really needs to include all the ECs for that relevant ECTT section. We found a few situations where looking at Module 3 and looking at the justifications, what was identified by the applicant as critical for control, critical quality attributes, or the direct linkage there, did not show up on the PLCM, and there was no explanation of it. So we had to rectify that difference. As well, the PLCM should clearly state the reporting category when different than regional regulation and guidance, or be really clear that we're following regional regulation and guidance, and should do so in U.S. terms. There have been a few times where applicants have used the Q12 terminology, and we've had to go back and clarify specifically what do you mean for a U.S. filing. When you say notification moderate, does that mean CBE 30 or CBE 0? We need to be explicit so that the agency has confidence in what we're approving. In the same way, the PLCM, I had mentioned this earlier, needs to be clear about the facility identifiers where those ECs are being approved. Again, because that's going to drive FDA's assessment of the pharmaceutical quality system, which is really looking at which facilities will be implemented, but also depending on the level of flexibility proposed, that will tailor the depth of assessment of the pharmaceutical quality system. So where there is flexibility in the established conditions proposed or lower reporting categories than we normally expect, our assessors look more into those ICHQ-10 principles of change management, effectiveness, etc., to see if the support is there to maintain ECs post-approval. By and large, our interactions with applicants have been fairly positive, receptive to our information requests. Modifications that are necessary to the PLCM have been made. In some cases, for example, when asking for justification or clarification, applicants, I think, have withdrawn proposals for established conditions if they particularly weren't ready to justify them. And in the case of a prior approval supplement on such a short clock, but they have been the minority. Count them on one hand where these situations have happened. And happy to report that to date, we've not denied approval of an application due to Q12. So very good success across the board so far, and I hope we continue that and expand it to more application types and more stages in the product lifecycle. We are so supportive of Q12 and its benefits to pharmaceutical product quality management and the life cycle, for example, in being more agile and making changes to avert shortages or respond to shortages or increases in demand. that we have applied Q12 principles during the pandemic, for example, with container closure system changes. We continue to address common misperceptions and questions by engaging with the industry and other regulatory partners. And we've been supporting global implementation of the tools, for example, PACMPs, in various forums. We continue to support the ICH Q12 implementation working group. The ICMARA pilots had a big focus on PACMPs and the approach to risk assessment. And we continue to share our experience with other regulators to help lower the global activation energy when it comes to the adoption of Q12. And not to mention our linkage of Q12 to other related ICH guidance documents, for example, M4Q and Revision 2, but also looking ahead into ICH Q14, where we talk about analytical methods, and you can even see the DNA in ICH Q13 when it comes to continuous manufacturing. So with that, I'm going to wrap up. A huge thanks to the ICH Q12 teams across the world, whether it's the expert working group or the implementation working group. Huge thanks to the FDA, ICH Q12, EWG and IWG teams, some key members here. But you can see on the third bullet there, the network is much, much broader than that when it comes to our support teams, our ECCC teams, and our Q12 assessment and implementation teams. The thanks cannot be repeated enough. With that, I'm going to wrap up, and I'm happy to engage with you further. Thank you very much.
Thank you for the great presentations. This takes us into our final break for 10 minutes. Please enjoy your break. Welcome back from the break. We're on the home stretch now, right into our next presentation on how nitrosamine research studies inform FDA on potential strategies and BE approaches by Dr. Dongmei Liu, who's a pharmacologist in the Office of Policy for Pharmaceutical Quality in OPQ. Next, we'll learn about the symbiotic relationship to ensure quality between USP and FDA by Dr. Pallavi Nithyanandan, who's the Director in the Office of Policy for Pharmaceutical Quality in OPQ. And finally, before our Q&A panel, we'll receive updates on product-specific guidances by Dr. Xiaoming Xu, who's the Division Director in the Office of Testing and Research in OPQ. Now, please join me to welcome our first presenter, Dr. Dongmei Liu.
Hello, my name is Dongmei Liu. I'm from Office of Policy for Pharmaceutical Quality. Today, I'm honored to have this opportunity to talk about nitrosamine research studies inform FDA on potential strategies and bioequivalence approaches. These slides talk about the importance of pharmaceutical quality. The outline includes the challenges of NDSRIs in drug products, and secondly, FDA-sponsored research project to support reformulations of impacted products, and lastly, FDA's considerations based on the research results. Nitrosamine issues has evolved very rapidly. There are two classes of nitrosamine generally. First class is a small molecule nitrosamine such as NDMA or NDEA. In general, they have a relatively simple chemical structure, and they are not related to the chemical structure of the API. It has been found in many drug products. NDSRI is another class of nitrosamines. They share the structure similarity to the API or the API fragments. NDSRI are formed during a drug product manufacture or during a shelf life or storage conditions. APIs with secondary or tertiary amines are exposed to the nitrosamine compounds such as nitrite impurity in the excipient, and they can form NDSRI under the acidic conditions. Many drug products are impacted by nitrosamine impurities. It is very challenging to determine the acceptable intake limits for each NDSRI because each NDSRI is unique to each API or the API fragment, and there's not much data available on the mutagenicity or carcinogenicity. FDA has recommended 3 potential mitigation strategies for NDSRI back in November 2021. That includes the first one is to screen excipients for nitrite impurities, and the second is related to the reformulation by adding small amounts of antioxidant. Some research paper has indicated that some certain antioxidants can inhibit nitrosamine formation. And the third is also reformulation by adding pH modifiers in the formulation, because nitrosamine formation is very favorable at the acidic conditions. If a basic pH modifier is added into the formulation, the microenvironment will be changed in the drug product, then potentially it could inhibit the NDSRI formation. FDA also encouraged manufacturers to come up with innovative approaches to resolve the nitrosamine issues. In order to evaluate the roles of antioxidants or pH modifiers in the solid oral dosage form, FDA has conducted or sponsored several research projects related to NDSRI mitigation. The first project is related to the quality. It is to investigate the risk of NDSRI formation in finished products formulations with excipient in solid states. And it's also studied NDSRI formation inhibition effect of excipient in drug formulations. FDA also sponsored 2 bioequivalence projects. The first project is a contract project by Pharma. It is to study the impact of antioxidants on in vitro permeability of some BCS3 APIs. The second bioequivalence project, it is as a CSER grant. and it is to study the effects of antioxidants on intestinal drug transporters using the in vitro method. Next, I'm going to provide more detailed information about these three projects. Project number 1, effectiveness of antioxidants in selected model drugs, mitigation strategy and impacts of reformulation on stability. Bumetanide was selected as the API. Along with the other excipients, it was formulated as a tablet formulation. 3 antioxidants were selected: ascorbic acid, caffeic acid, and ferulic acid at the different concentrations up to 1% of the weight of the formulation unit. And 2 kinds of pH modifier are used. They are acidic pH modifier or a basic pH modifier by using sodium bicarbonate. In the wet granulation procedure driving steps, 60 degrees of heated air was used for this purpose. The different formulations were put into the stability chamber under 3 conditions. They are 50-75 for up to 1 month, 40-75 for 6 months, and then 25 to 60% of relative humidity for up to 6 months. The figure and table in this slide demonstrate the inhibition efficiency by the antioxidants and the pH modifiers. And it was demonstrated as this 6 months stability under 2 conditions, 40-75 and 25-60. Among the antioxidants, ascorbic acid showed the strongest inhibition efficiency among 3 of them. And it's followed by the caffeic acid and then ferulic acid. With the increased concentration of antioxidants, the inhibition efficiencies also increased. For the pH modifier, the basic pH modifier by sodium bicarbonate showed the overall the strongest inhibition efficiency for this study. So the take-home message for this study right here is that small amounts of pH modifier, especially the basic pH modifier or some certain antioxidant, can inhibit NDSRI formations. Next, I'm going to talk about the 2 research projects on the bioequivalence, because there are challenges in the bioequivalence for the products reformulated due to nitrosamines. As we all know, most of cases get impacted by the nitrosamines are in solid oral dosage forms. According to FDA SUPAC guidance, the addition of new excipients belong to level 3 changes. Typically, in vivo B study is recommended. But for the in vivo B study, it is going to cost a lot, and also it spends extensive time to complete the studies. At the same time, FDA expects manufacturers to resolve the nitrosamine issues as soon as possible and complete supportive study within certain time frame. In order to explore whether there are some other bioequivalence approaches for this purpose, FDA sponsored 2 research projects. The first bioequivalence project is on the effects of antioxidants on intestinal permeability of BCS3 drugs. BCS3 drug substance was selected because of their feature of a high solubility and low permeability. That means if there is some variation in the permeability, that would be the worst case of a scenario for the BE to be impacted. 4 antioxidants were selected. They are alpha-tocopherol, ascorbic acid, cysteine, and propyl gallate. The maximum amount of 10 mg of antioxidants was selected because that was based on the calculation of a dosage units of 500 mg and give up to 2% of the weight of antioxidant. The IDAS system, in vitro dissolution absorption system, were used in this study. Basically, the CACO2 cell monolayers will grow on the snap well and then inserted into the permeation chambers. These 2 chambers were inserted into the dissolution vessels. The atenolol was used as an internal marker in this study. In these slides, the figures show the typical permeability observation for BCS3 drug substances when co-dosed with antioxidants. For this case, it is acyclovir. When it's co-dosed with alpha tocopherol at the different concentrations, the permeability has no significant difference from that without the co-dosed antioxidant. And in this second figure, that is atenolol permeability, it demonstrated that the cell monolayer has no integrity issue during the testing. The apparent permeability was calculated as a mean and the standard deviation. And this table summarizes the results of permeability from a different drug substance when co-dosed with different antioxidants at different concentrations. So basically, the take-home message is that the tested amounts up to 10 milligrams of this tested antioxidant do not have a significant impact on the model drug BCS3 drug substance permeability. The second bioequivalence project is on the effects of antioxidants in drug products on intestinal drug transporters by UCSF. As we all know, some nitrosamine-impacted products are substrates of intestinal transporters, such as PGP, BCRP, OATP2B1. First two are efflux transporters, and the last one is influx transporter. And the membrane vesicles, which overexpress the intestinal transporters, are used for this study. There are 4 steps involved in this study. Step number 1 is creation of a library of 30 antioxidants used in the oral formulations, 22 of them from FDA IND database and 8 from a natural product. Step number 2, each antioxidant at a concentration of 200 micromolar will be studied for the inhibition of the transporters. Step number 3, if the inhibition effect was observed for more than 50%, the IC50 of the antioxidant will be estimated. Step number 4, the IC50 of the antioxidant will be compared with the estimated intestinal concentration, just try to see whether there are any potential drug-drug interaction. The probe substrates in this study are listed in the table below. And to the table right, it gives the information of the uptake percentage of substrate when co-dosed with the antioxidant. The antioxidants listed in this table are from FDA IND database, which also showed no significant impact on the transporter activities. However, there were 2 antioxidants that show the inhibition effect to some certain transporters. They are ascorbyl palmitate and butylated hydroxyanisole. Then the IC50 value of these 2 antioxidants to the certain transporters are estimated and labeled right here. After that, the maximum intestinal concentration I got was calculated as well. Then the ratio of the IGAT over IC50 value was calculated and also compared with 10. Because according to the FDA guidance on the drug-drug interaction, if this value is more than 10, then that means that there will be a clinical significance. But for these 2 antioxidants, for all the scenarios, and the values are all less than 10. So that means these 2 antioxidants are unlikely to inhibit the transporter activities. As a summary, the project results support the proposed nitrosamine mitigation strategies and also inform other bioequivalence approaches. The quality project with the reformulations with the pH modifiers and the antioxidant indicated that these 2 reformulation strategies are valid. Small amounts of added excipients can effectively inhibit NDSRI formation. For the reformulated drug product, and for the BE considerations, the in vitro bridging studies such as permeability or transporter activity study have great potential to demonstrate BE. The studied antioxidants at tested ranges didn't have any significant impact on the in vitro permeability of BCS3 drug substances or intestinal transporters. but more supportive data are needed for antioxidant levels beyond those researched, or other type of antioxidants are used in the reformulation. Manufacturer can also consider some other B approaches such as PBPK modeling or IVIVC, in vitro in vivo correlation. Manufacturer can also use different approaches for low risk versus a high risk product, but need to provide sufficient justification for this risk-based approach. FDA encourage manufacturers to come up with novel approaches. If under this scenario, please contact FDA for further discussion. At last, I want to indicate that FDA continues to consider different approaches for products impacted by NDSRIs and nitrosamines. With all the efforts, FDA wants to help industry to resolve the nitrosamine issues as soon as possible and to ensure the safety and efficacy of the medications to the public health. I want to acknowledge my senior colleagues from Office of Pharmaceutical Quality, Office of Generic Drugs, and Office of New Drugs. We have been working on that since 2018 and has a lot of progress. I also want to thank FDA OPQ OTR Lab, PharmaRun, and UCSF Dr. Giacominni's lab for conducting these 3 research projects. And thank you so much for your attention.
Hello, my name is Pallavi Nityanandan and I serve as the Director for the Compendial Operations and Standard Staff in the Office of Policy for Pharmaceutical Quality. Today I'll be presenting about FDA USP collaboration, our working relationship and partnership in the development of USP standards. Today I'll be presenting about the overview of FDA USP interactions as they pertain to the development of standards, then talk about the key mechanisms through which FDA interacts with USP. One is through the government liaison program and then through our active review and comment process on standards proposals published in the Pharmacopoeia Forum. And we'll then discuss the important role industry plays in the development of USP standards, and then we'll take any questions after the presentation. FDA has had a long-standing collaborative relationship with the USP. FDA values engagement with external organizations that develop standards because standards enable consistency, predictability, credibility, and lead to better science-based decisions. USP is legally recognized in the Food, Drug, and Cosmetic Act under Section 201 as the official compendium of the United States. Both USP as well as NF, the National Formulary, or any supplement to them has been recognized in this section. The USP and NF official standards for strength, quality, purity, identity, packaging, and labeling can be used by the agency to support any charges of adulteration or misbranding, which we will take a look at more closely in the next upcoming slides. The adulteration provision comes from Section 501B of the Food, Drug, and Cosmetic Act, which states that if the strength, quality, or purity of a product that falls under an official USP monograph differs from the requirements of that standard, then the differences have to be plainly stated either on the product label or in the labeling. However, the identity requirement in the monograph is mandatory and there is no option to label out of it. The misbranding provision comes from Section 502 of the Food, Drug, and Cosmetic Act, where a drug or device will be deemed misbranded if it is not labeled with the official established name, which is the monograph title for a drug substance, drug product, excipient, or even a compounded drug. The established name is the monograph title, and also the requirements for packaging and labeling must be met, anything that's outlined in the monograph. There are some nuances for packaging, but we won't get into that right now. As I mentioned earlier, one of the key mechanisms by which FDA participates in USP standards development is through the Government Liaison Program, by which FDA subject matter experts are allowed to serve on USP standards development committees. FDA representatives, mainly subject matter experts, serve on USP committees and panels, and currently there are about over 130 liaisons or FDA staff who serve in this government liaison role. And in addition to CEDAR, there are also government liaisons from various other FDA centers, such as the Center for Biologics and Center for Veterinary Medicine, Center for Food Safety and Nutrition, Center for Devices, as well as the ORA, which is our Office of Regulatory Affairs. And their primary role is to provide input on behalf of FDA to enable better alignment between FDA's current regulatory thinking and USP standards in development, which we hope will eventually provide better clarity for stakeholders on various topics. And because the topics are very broad in scope, they may need to bring back some of the information for internal discussion within FDA as permitted. And the overall program is coordinated by COS, because the main purpose behind the coordination is for recruitment training, pre-meeting, and also staying in touch with the government liaisons to advise them on policy and enable better collaboration between various teams as we keep track of the overall progress on various topics. The other important way in which FDA participates in USP standards development is through the active review and comment process for articles published in the Pharmacopoeia Forum. So we do have an active process of reviewing Pharmacopoeia Forum proposals and providing comments back to USP within the designated comment deadline. So when a proposed standard is published in Pharmacopoeia Forum, we do an initial triage and determine who needs to perform the review and how widely it needs to be distributed within the organization, if we need to collaborate with other centers outside of CEDAR, and then we take the appropriate steps to send out these requests for review or perform our review internally. And a lot of times when we perform the review internally within COS, we still get the opinion of others by sharing the review. Or there could be some cases where it is a proposal that's already gone through a revision cycle and there is just one aspect that's being addressed and we are well aware of what that is, and we may choose not to send it out for extensive review. So when we get the comments back, we draft the responses, and a lot of times when it's a new general chapter or some complex topic, then it involves meetings, getting together various offices. Like we do have some different perspectives that need to be aligned and consensus reached before we send the comments back to USP. So then we will follow up through our government liaisons on discussions of those comments, and the government liaisons may be able to provide further elaboration on the written comments sent on the PF. While we have a robust and collaborative relationship with USP, there are also some unique challenges with the review and comment, especially when it comes to monograph and specifications included therein. So because USP is a non-governmental entity, we are not at liberty to disclosing specific criteria or impurity names, etc. As when we comment on the PF proposal. So our comments tend to be quite general, and then there has to be some follow-up that USP does with the industry, and then if they get additional information, they can confirm things with us, but we cannot give them the specific and the direct information needed as to what the range should be or what impurities should be included, etc., because they are considered as company confidential trade secret information. So that's how the process works, and there is a lot of email, both pre-PF and also post-PF. There are a number of communications between FDA and USP just to check on specific aspects. This is a summary of how the interactions work between FDA and USP for monograph development. When USP prepares a monograph proposal, they would share it with the government liaisons for their comments, but then the more detailed reviews happen during the PF stage, where COS coordinates review with the monograph and with the review divisions within FDA, and then we collate the comments and send them to USP, and then they would evaluate our comments along with other comments from stakeholders, and then they will propose further revisions based on the additional information received, and that new proposal or revision to particular aspects of the monograph proposal will be shared by email, and COS provides feedback either which is mainly involves just concurrence. Again, we don't disclose any specific information, and then USP finalizes the proposals as official standards. USP monographs are important to both FDA and industry because they are not only applicable to previously approved applications, but could also significantly impact a pending application's review. And a good monograph certainly improves the efficiency of the process by providing information for product development, which could be very useful for the applicants, especially the generic drug developers, and also when firms follow USP method and acceptance criteria, there is generally more abbreviated requirements for verification or demonstration of suitability for use as opposed to a full-blown validation. We have also been engaged with USP since 2010 on modernizing outdated monographs, for which they have done a great job and updated many of the monographs, because this is really important, because if the outdated specifications are relied upon by applicants, then it just slows down the review process and also can be misleading to firms. They have spent time using something that may not be acceptable to the agency. Now that you have heard about FDA's process for reviewing USP standards proposals and some of the limitations we face, and now I would like to move on to emphasize the role of industry, the important role of industry in contributing to a revision of USP standards. So applicants, DMF holders, and manufacturers are recommended to have a robust process for reviewing and commenting on USP monograph proposals that get published every 2 months in Pharmacopoeia Forum and any other notice of intent to revise, which are posted that give you early advance notice of an upcoming revision. And also, please consider your data, your batch data while commenting. If your data indicates that your product can meet what's being proposed in the monograph, then there is no need to petition USP for a wider acceptance criteria because you are already meeting what's being proposed. You could just update your specifications at the next opportunity. And contributing improved analytical procedures to USP enable monographs being up to date, so they are beneficial to public health. This is to summarize some of the major ways in which FDA interacts with USP. We have an active role in the review and comment of USP standards proposals, as well as the monthly nomenclature ballots, which is mainly through the participation of our government liaison to the nomenclature and labeling expert committee. And there are multiple emails on a daily basis from both on prior to publishing a PF proposal as well as post PF inquiries. And we talked about the government liaison program, which is managed by COS, and COS staff also participate as GLs to some of the committees and attend as observers as needed for some of the policy issues. Besides these, there are also some other routine engagement on broader policy issues between COS and USP, where we also bring in other pertinent subject matter experts and FDA staff as needed for topics that impact more than one monograph, for example. Additionally, there are also engagements with industry and other stakeholders through USP. And we have a FDA USP quarterly meeting, which involves representatives from various FDA centers as well as leadership. And there are also some one-on-one or other engagement between senior leadership of both organizations, and USP also plays a role in the USP convention, which happens every 5 years. We have 6 delegates from various FDA centers, and the commissioner's office. We play an active role in submitting resolutions for USP to consider adopting, as well as provide feedback on resolution proposals. And throughout the cycle, there are also activities of the Council of the Convention, which is one of the governance bodies of USP, and participate in the nominating committee for the Council of Experts when it's time to prepare for the new 5-year cycle. Last but not the least, we also engage in pharmacopoeial harmonization, serving as a partner to USP in the PDG process. In summary, FDA very much values our collaboration and partnership with USP, and we believe that the standards serve public health by providing minimum legal standard and standardized quality and purity requirements for drug products across manufacturers, which are developed through an open process that provides for broad stakeholder input. And also we have different regulatory pathways for OTC over-the-counter products and prescription products, and monographs provide equalized standardized quality and purity requirements for drug substances and for drug products where monographs exist. And USP standards serve as effective tools that can be used in FDA review and enforcement activities, and for certain complex products such as the biologics, monoclonal antibodies, etc., FDA supports non-monograph standards and performance-based standards, which is the direction USP seems to be moving in. Thank you so much for your time and attention. I'll be glad to answer any questions you have at this time.
Good afternoon. I'm Dr. Xiaoming Xu. I lead a group of researchers in the Division of Product Quality Research in OPQ. In the next few minutes, I'll briefly talk about GDUFA research and specifically focus on the translation of the research findings to support the development of product-specific guidances, or PSG. And I'll limit the scope of the discussion to complex products, as they are often more challenging to develop and hence more likely of needing research. The GDUFA program was first enacted in 2012, then reauthorized twice in 2017 and more recently in 2022 under the GDUFA 3. And in addition to aligning the user fees and review timelines, the GDUFA program also allocates resources to the FDA to fund research to facilitate generic development and review. Since 2013, FDA has awarded over 200 contracts and grants, as well as conducted numerous internal research projects led by FDA staff. This research facilitated the development of new tools and methods which helped FDA and industry to evaluate the generic drug equivalence. A key outcome of this GDUFA research is to enable more efficient development and assessment of generic drugs, as well as improve resources for the development of PSG recommendations. And a key aim of the GDUFA research is to disseminate the research findings to improve the general understanding of these products and to aid the generic industry in their development program. Therefore, results from these researches are shared through scientific meetings as well as peer-reviewed scientific publications. The Product-Specific Guidance Program, which began in 2007, aims at facilitating the development and approval of safe, effective, and high-quality generic drug product by providing FDA's current thinking and recommended approach for demonstrating equivalence for a specific reference-listed drug product. These PSGs are published on a quarterly basis on the PSG website, as shown below. To help the industry develop complex generics, as negotiated in GDUFA 3, FDA has agreed upon priorities and goal dates on the development of the PSGs. Specifically, starting October 1st, 2022, a PSG will be posted for 50% of the complex new drug applications within 2 years after the approval, and for 75% of the complex NDAs within 3 years of the approval. And these are very aspirational goals, especially considering the challenges of the complex drug product, which I will explain later. Now, to meet this new goal, we will need a process that is very efficient in identifying and addressing any potential complexities and hopefully initiate any needed research early in order to meet the goal. Now, let's look at some of the challenges in developing PSG, especially for complex products. On average, there are close to 100 NDAs approved every year that may warrant the development of a PSG. Of those newly approved NDAs, about 25% are considered to be a complex product. As we have seen with some recent NDA approvals, the complexities can vary greatly between products and across disciplinary areas, and can benefit greatly from a close collaboration across multiple FDA teams. The use of new materials, technology, and processes in these new drug applications also highlight the potential scientific gaps and the need for new and reliable analytical methods, which needs new research and innovation. And furthermore, in GDUFA 3, the new goal set on the PSG development of the complex products may pose additional time constraints, especially if additional research is needed. So early engagement of research and review will be very crucial. And lastly, taking a life cycle approach towards the development of the generics necessitate a better communication and a collaboration across teams and also with the industry as well. I will use one example to illustrate the importance of starting research early. As you may know, cyclosporine ophthalmic emulsion approval in early 2022 was a very notable example of the impact of the GDUFA research. Showing on this slide is a timeline of the approval overlapping with research as well as the PSG development. Research generated supporting data which allowed for assessment of product-specific quality attributes, best measurement practices, and assessment criteria to support the establishment of bioequivalence. However, it took nearly 10 years from the original NDA approval to develop scientific recommendations, and another 9 years to translate that to first generic approval. Needing such a long time to approve one drug are rare, but it does highlight the challenges with the research, especially the time that it may take, as well as the need to timely translating research findings into regulatory actions. And the question that we have asked is, how can we speed things up and doing it more efficiently? Prior to the GDUFA 3 rollout, FDA has established a new internal program to identify and direct research to support the complex PSG development. There are 10 subject matter expert teams specializing in various complex areas, with members scattered across 9 CDER offices. Within the last 2 years, this pilot program has conducted 59 triage meetings on newly approved complex NDAs, and already identified 8 that need additional research before a PSG can be developed. Now, briefly, a few more details about this program. It is established specifically to support the complex product PSG development. It includes relevant subject matter experts from both OGD and OPQ. And most notably, the program connects research staff directly with regulatory assessors as well as policy groups to discuss challenges and the needs for each newly approved complex NDA. The teams are created based on complexity areas such as ophthalmic, inhalation, and dermatological, so each team is very familiar with the topic area. Each SME team is tasked with making decisions on complexity and gaps to the PSG development and approval. If a gap is identified, the team also decides on the objective of the research. Afterwards, a dedicated research project is created to generate necessary evidence and to fill the knowledge gaps. And this could be a research project conducted internally within the OPQ lab, or could also be awarded externally through the GDUFA research program. We started piloting this program in 2021, and it became fully operational from the start of the GDUFA 3. As mentioned earlier, including the pilot phase, we've already completed 59 triages and already initiated 8 research projects. Here is another view of the program in terms of the timeline. As we know, research can take time, so it is important that we start triaging the NDA product soon after the approval, and through the triage process to narrow down the products, especially the complex ones that can benefit from targeted research. After we complete the research, knowledge gained during the research will need to be translated to recommendations, which will then be put into the PSG. And the example of the recommendations may include new methods or new approaches to demonstrating bioequivalence. I want to provide 2 examples of the SME triage. One is an ophthalmic emulsion of cyclosporine, and this product was approved in June of 2021, and the triage meeting was conducted in October of 2021. At the triage meeting, the SME team collectively decided that there is sufficient understanding from the previous research and the precedents, and therefore recommended immediate development of the PSG. As you can see, the PSG for this product was published in August of 2022, just 14 months since the NDA approval. Another example is a little bit more complex. This product is an ocular insert of dexamethasone with sustained release properties. The NDA was approved in November of 2018, and the decision from the triage team was that research is needed to better understand the product before we can recommend a BE recommendation. The identified gaps are primarily in the area of material characterization and in vitro measurements, and this work is currently ongoing, and we hope to complete it soon and share the findings with the public. As part of the GDUFA 3 commitment, the agency aims to further improve the forecasting of the PSG publication, which includes both the non-complex and complex products. And this helps the agency communicating the priorities as well as the timelines of the PSG development, which allows the industry to better plan their generic drug product development program. This forecasting list is updated every quarter, and the link is provided below. As you can see, GDUFA research has a very significant impact not only on the ability of the FDA to develop product-specific guidances and to support the pre-ANDA program as well as the ANDA assessment, but is also a valuable resource for the generic industry. In closing, Product-Specific Guidance Program provides FDA current thinking on the type of studies and information to support the development and approval of safe, effective, and high-quality generic drug product. GDUFA research plays a critical role in the generation of evidence and knowledge and supports the timely development of the PSGs. I want to thank a few people who helped me putting together the slides, and also my colleagues at OPQ and OGD in contributing to the success of the PSG program. And thank you for your time and attention. Thank you.
Thank you all for the great presentations. At this time, we'll remind our presenters and panelists to attach their mics and turn on their videos for our live Q&A session. And as a reminder to our attendees, if you haven't had a chance to present your questions in the Q&A chat pod, please do so now. We'll answer as many questions as time allows. We do have some panelists that are connecting the video and coming in. We'll give them just a few moments for that to take place. All right, we do have more panelists coming on board. I do have 2 up, and I think we're about 3 short as we're waiting here just a moment. And if you haven't submitted those questions, if you have your favorite one coming in, please, now is the time to submit them. All right. It looks like we have just about our entire panel there. We're going to move into our panel session. Our first group of questions will be for Layla Weezer. And here is the first question. Is there a single website from the FDA that describes all current guidance and compliance requirements for quality issues?
Hi there. Thanks so much for the question. I think that's a great one. Where's all the information at that we speak of? So something that I just wanted to share with you all was on the FDA, the external internet page, www.fda.gov. If you go on there, there's actually an entire section. It would be drugs, and then there's a section that you can click on with a left navigation for guidance, compliance, and regulatory information. And it will actually take you to guidances and drugs. From that page, there's actually a whole search option as well, and it would be by topic area. So some folks have asked also about quality specific documents. One of the drop downs, in fact, is pharmaceutical quality. But there are a number of other options as well, which you'll be able to see on that page. So again, if going to the home page, going to drugs, and then you'll be able to select guidance, compliance, and regulatory information. And go ahead and click on guidances and drugs, and you'll be able to then find the search function for that. Thanks so much.
Thank you for responding to that question. I've got all 6 cameras on the screen right now. I want to take this opportunity to welcome to our panel Susan Zuck, who's the Branch Chief, Division of Regulations, Guidances, and Standards in the Office of Policy for Pharmaceutical Quality and OPQ. Moving back to Layla Weezer, we do have a couple more questions. And here is the next question. Is there a table that links the regulations, guidance, and MAP or compliance program documents on the FDA website?
And thanks again for another good question. I think that really ties back to the conversation that we had about the regulatory framework. So how does everything connect together when we're trying to understand the story? And to answer that, there's not a table per se, but that same website I was mentioning to you, it's a one-stop shop for those resources. So in addition to those guidances, and again, it's by topic, we also have the link that takes you then to the external facing maps that are available, as well as the compliance programs that are available. Looking at it, the search function by topic, we've done our best then in that regard to make it user-friendly by topic. So you're not needing to look in multiple places for the policy documents. You can look at it by topic and by document type. So my suggestion, take a look at that page that I had mentioned earlier, explore, and it should be able to tell you that story by document type, but not only that, by topic, more importantly. So thank you.
Thank you for responding to that question. We have one more question for Layla Weezer in this round, and this is the question. Can you elaborate on a, quote, draft guidance is not for implementation, since FDA inspection staff and review staff quote draft guidance documents in 483s, CRLs, and IRs?
Thanks so much. So for that one, there was actually a number of questions. I want to make certain you know that we hopefully address this with the one response, but that was a great example of the type of question that we were receiving about this. So just for clarification, draft guidances, while they're not final, they do provide insight into how we're interpreting our regulations and statute. So the basis for actions that FDA takes are ultimately pointing back to statute and regulation. So with non-binding guidance, it's just that. It is non-binding, but alternate approaches can be used. But we just want you all to be mindful that while a guidance is a guidance, it's providing the interpretation. There may be elements in a guidance that are directly pointing to requirements in a statute or regulation, and you'll be able to denote that because we have quotes or we have it sourced to what statute or reg it's pointing to. So there's really that opportunity. It's clear when you're reading it then, but we just want to make that clear. So that should have answered a number of questions in this space. So thanks again for the questions so we can provide that clarity to the rest of the audience. Appreciate it.
Thank you for responding to that group of questions. Moving on to our next panelists, we've got a few questions that came in, one big one for Commander Mahesh Ramanathan. And here is the question for Commander Ramanathan. At a recent RAPS convergence meeting held in Montreal in early October, the experience shared by one presenter indicated that regulators, and this comments pertain to the FDA and Health Canada, are defaulting to regulatory categories as defined by the regs, except for very low risk changes. If you can provide additional feedback on what frameworks may be in the works to enable greater adaptability of the regs as regards an ICH Q12, it would be appreciated.
Thank you for the question. That's a very good question, in fact. I think, speaking on behalf of FDA and our experience in CEDAR when it comes to the implementation of Q12, what we've seen is actually the opposite. We've seen applicants come in with proposals with flexibility to the reporting categories with appropriate justification. And when they are appropriately justified, as I showed on the slide, we'd approved nearly 15 or so. The situations where we were unable to reach alignment, the applicants were not really in a position to provide scientific justification for the proposals. So it wasn't a situation where we just couldn't see eye to eye on the proposed reporting category. It was really the science wasn't there to back it up. But I am happy to say that when the science is there, we've found ourselves in the position to really take advantage of the flexibility in the US framework. Looking at 21 CFR 3.270 or 6.112, the regs are built to inherently have a lot of flexibility in what could be considered any one of those particular reporting categories. So my feedback here is really, the extent and the advantage that an applicant can get is really going to come down to how strong the science is and how well they can present in their application. As far as frameworks, I look forward to our ongoing collaboration with other regulators. For example, the ICMA pilots, where we're thinking about ways where we can align on how we approach risk and how we look at particular changes, for example, with change management protocols. So I'll stop it there and give it back to you, Jeff.
Thank you, Commander Ramanathan. We're going to move on to our next panelist. We only have about a little over 10 minutes left. We have a question that came in for Dr. Dongmei Liu. And here's the question for Dr. Liu. What do you think this approach will affect BCS 2 or 4?
All right. Thanks for the great question. Here, we just talk about the oral drug products, and so BCS2, as you all know, that has a high permeability and low solubility. So this in vitro bridging approaches, of course, you can apply to this BCS2 drug product, but you need to consider that because it's a high permeability, so likely the addition of a small amount of antioxidants may not have a significant impact on the permeability or absorption. But in terms of the solubility, whether there are any kind of impacts on its solubility or dissolution, and because of the BCS, it has a low solubility, so you need to consider and have a good test and to make sure the solubility is not, and the drug release is not impacted. And in terms of the BCS4 drug product, they are low solubility and low permeability drug products. So they are considered as high-risk drug products. In general, for these high-risk drug products, we would want to see more supportive information to support the formulation changes. Thanks.
Thank you for responding to that question. Moving on to our next panelist. We've got a question came in for Dr. Pallavi Nithyanandan. And here is the question. Do USP monographs apply only to generic drugs and or drug products labeled USP?
Thank you so much for the question. The answer is no. If there is a USP, applicable USP monograph, it applies to all marketed drug products in the US, and even products that fall under the OTC non-application system, as well as unapproved products would also fall under the monograph requirements. Thank you.
Thank you for responding to that question. Moving on to our next panelist. We do have a question that just came in for Dr. Xiaomin Xu. And here is a question for Dr. Xu. Since Q13A is considering harmonization of BE requirements, will FDA adopt Q13A instead of PSG?
Yeah, thank you for the question. I'm interpreting the question about Q13A is referring to the M13A for the bioequivalence for the immediate release drug product. So short answer is they serve different purposes, product-specific guidance, outlining FDA thinking about the development, including not only the bioequivalence, but also pharmaceutical equivalence approaches that a generic company should adopt or recommended to consider during the development phase. So M13A is a general approach guidance across multiple regulatory agencies outlining thinking about the BE specifically for immediate release formulation. So what I was describing in the talk, emphasis more is on the complex formulations, which is really not covered under the M13A. So thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions on nitrosamine that came in for Susan Zuck, and here is the first question. A new FDA guidance on nitrosamine impurity categories was published in August 2023, and there is also an FDA webpage on possible ND SRI AI categories. Does FDA intend to periodically update the website of possible ND SRIs?
Okay, good question. Thank you. I hope my sound is good. Yeah, that guidance just published in August, and it had an accompanying website with AIs. As the guidance and the website say, we know that this is an evolving science, and we do intend to periodically update the AIs on that website. So keep checking.
Thank you for responding to that question. We do have a couple more questions for Susan Zuck, and here's the next one. Again on nitrosamines, the nitrosamine guidance dated February 2021 R1 indicated that FDA accepts LOQ at or below 0.03 part per million. Does this apply to all nitrosamines for various products?
Okay, thanks for that question. Actually, FDA has been asked about that statement in the nitrosamine guidance quite a bit. When we published that guidance, we didn't have a lot of AIs for products. Of course, now we have this new guidance with far more AIs, and the AIs are higher than what we had in the original guidance. In that original guidance, we had published the suggested LOQ of 0.03 to let industry know that they didn't have to go very, very low with their LOQ. We knew that the methods had to be very sensitive, and if the LOQ had to be extremely low, that would be impossible for many manufacturers. That being said, now that we know that we can have some higher AIs for NDSRI products, for example, limiting an LOQ to 0.03 is not necessarily appropriate, and it certainly isn't necessary. And so in thinking about this, the agency is thinking that it might be best with those higher AIs to set the LOQ based on the AI itself. That's our target analyte, and that's our target limit. So a low LOQ, that would be practical, consistent with Q2, ICH Q2 guidance, would be something around perhaps 10% or lower of the AI, and that would be an acceptable LOQ for some of those high AI products. Thanks for the question.
Thanks for responding. We do have one more question for Susan Zuck, and here it is again on nitrosamines. If any relevant ND-SRI impurity cannot be synthesized, then this could be an indication that either does not exist or there is no risk of it being formed. In such cases, is it okay to skip the confirmatory testing?
Thanks for the question. I believe this question is probably referring to the hypothetical list, table 1 in the new guidance. Because some of those NDSRIs are hypothetical, we know that it's possible that when an applicant does a risk assessment and tests for this impurity, they're going to find that maybe it doesn't form. And we would encourage applicants to actually see if they can form the NDSRI by stressing the API, putting it under those conditions that would normally form the NDSRI, for example, combining it with a nitrite ingredient in order to try to force it to form. If it can't form, and if the applicant can show us that they have done their due diligence, that they have taken experiments to try to form the NDSRI, then yes, it would be appropriate not to develop a method and routine testing for that NDSRI, but the applicant should show the agency that it really can't form in the product.
Thank you for responding to that question. We do have one more question. We've got just a little over 2 minutes left. We'll try to get a couple more in for Commander Mahesh Ramanathan. And here is the first question. Are the use of established conditions and reduced reporting categories limited to well-understood small molecules or open to novel regenerative medicinal products? And in addition, would manufacturing processes that use advanced technologies such as AI also be eligible?
Thank you for that question. So in the context of scope and applicability, Q12 is applicable to small and large molecule products that are regulated by either the Center for Drugs or the Center for Biologics. So that would include your regenerative medicines as a potential example there, and drug device combination products. As far as applicability, as far as like knowledge or controls, I think it's important to remember that Q12 is available to applicants of all applications, whether that's innovator products or generic products. And they're not limited to those situations where, someone is doing very advanced development or very advanced manufacturing. It can be equally employed to generic products where, an applicant might be relying more on platform knowledge for products they've already manufactured or that they've gained for that particular drug product. It's extremely flexible to come in and propose established conditions for a particular application product. Doesn't matter what kind we're talking about. So let me pause it there and give it back to you, Jim.
Thank you for responding to that question. We have just under a minute left. We're going to try to squeeze one more in with Dr. Xiaoming Xu. And here is the question. For PSG development and publication, will FDA publish an official PSG while the RLD company against it with lots of difficult questions?
Yeah, thank you for the question. We always receive difficult questions at FDA. So, I think the short answer to that is the purpose of developing PSG doesn't mean that FDA is ready to review or approve the application. It simply means that
will provide the recommendation to the developers outlining the approach that they should take to develop the product. So we oftentimes receive the feedback from the public. Sometimes can be from the ILD companies, but also sometimes can be from another company proposing alternative method. Again, the PSG doesn't mean approval. PSG doesn't mean those are exact recommendation that company need to follow. And the company still feel free to develop alternative approaches as long as it can be can be provided in their application to justify their proposed approach. Yeah, hope that answers the question. Thank you.
Well, that's all the time we have for questions in this final Q&A panel for today. A huge thank you to our presenters and panelists for answering numerous questions that came in today. It's now our pleasure to introduce Dr. Adam Fisher, who's the Director of Science Staff in OPQ, to provide us with some closing remarks. Please join me to welcome Dr. Fisher.
Thank you so much, Ray. And thank you to the policy panel who just wrapped up, who gave us so much great information. Even though I work in this area every day, I just learned a few new things. I am Adam Fisher, Director of Science Staff, and I thank you all for joining us virtually here today in what I believe to be the premier global event focused on pharmaceutical quality. We hold this event every other year, and we take planning it very seriously, trying to produce the best topics and speakers for our audience. And I hope you will join me in thanking today's speakers and panelists for their time and effort in making this event possible. Without them, we could not have done this. We started out today hearing from the FDA Commissioner and the Director of CDER's Office of Pharmaceutical Quality about the importance of what we're addressing here. We learned about quality assessments, including new technologies for assessment and the latest in evaluating facilities. Then we went in on all of the latest in pharmaceutical quality policy. We had wonderful audience engagement in the Q&A today, which led to these robust panel discussions that I have really enjoyed. Now I ask you to join us again tomorrow. We'll start with a featured presentation on Project Orbis, which has really been a groundbreaking regulatory program, followed by sessions on supply chain and advanced manufacturing. If you're like me, you don't go more than a day without reading about one or both of these session topics in the press. So hear more about FDA's activities in these areas tomorrow. As a bit of a plug, I'll note that I'm speaking in the advanced manufacturing session tomorrow on a brand new development for the first time. I'm excited to see what tomorrow holds, and I look forward to engaging with you then. Please have a wonderful evening, night, or morning. Back to you, Ray.
Thank you, Dr. Fisher, for providing those pearls of wisdom for pharmaceutical quality. We do have a few housekeeping reminders. Please remember the 14-day deadline, that's November the 15th, to claim the certificate of attendance to support Cs for SOCR RAPS, SQA, and ACRP. As a special note, remember to download the certificate immediately upon creating the certificate. Otherwise, this opportunity will be lost. Recordings will be posted in approximately 7 to 10 business days. On behalf of CDER Small Business and Industry Assistance, we hope that you enjoyed today's presentations and found them helpful. We'll continue the discussion on pharmaceutical quality tomorrow at 9 a.m. Eastern Time. Thank you for attending, and we look forward to seeing you tomorrow at 9 a.m. Eastern for Day 2 of Pharmaceutical Quality. This concludes Day 1 of PQS 2023.